Welcome everybody to the second day of the Advances in Digital Media workshop. I am Austin Mast, and I've met many of the in-person people, but I realize that there are a few who are joining remotely. I'm one of the co-PIs of iDigBio. iDigBio is the NSF's national resource for advancing digitization of biodiversity collections. My domain is responsible for digitization, workforce development, what is now increasingly called participatory sciences, what has often been called citizen science or community science. I want to begin by thanking Nelson Rios and his team and his colleagues who are hosting us here at the Peabody Museum. It's fantastic. It's really an amazing place, and I encourage everyone who's joining remotely to make time in their schedule next time they're in New Haven to check it out. I also want to thank Jill Goodwin for her great work. Jill has taken this from idea to events, and that has big bonuses for our community, big opportunities for us. I also want to thank Kit Lewers and Summer Mangarelli for their help. They not only brought their expertise as participants in the workshop, but have also been lending a hand. Can I ask you, if you were involved in any of the planning meetings for this, can you raise your hand? Mike, Nelson, and others.
Yes.
Others who have been involved in this have been Hank Bart, Zach Randall, Gil Nelson, who's also involved in the framing of this series early on. I want to mention to everyone that we are offering a digital imaging for collections course that is July 9 to 12. It is through the Digitization Academy, and this is a 12-hour course. It's online, it's free. I expect that everyone in the room has this already under their belt, but you might supervise somebody who could benefit from this leveling up. One of our motivating questions for the workshop series is, how can we use media technologies to position biodiversity collections for even greater relevance to science, society, and Earth's biota in the future? This is the kind of question that asks us to lift our heads and think a bit about the relevance of our everyday activities. It's a question that helps us to clarify purpose, and purpose can lead to great things. This is the kind of thing that iDigBio does. iDigBio is a catalyst for further successes in our community. And as you all know from chemistry, the presence of a catalyst doesn't mean that there isn't an activation energy required. It just means that the activation energy is reduced. There's still a lot of heavy mental lifting to do. We get together as a catalyst. We get together a sometimes novel combination of subject experts and give them something to think about that supercharges the community for even greater relevance to the world. I learned a lot yesterday, and I hope everyone would agree that they learned at least a couple of new things relevant to how they do business. For those joining online, we had a pair of excellent tours of the state-of-the-art imaging facilities here, then had conversations around the group exercise. The group exercise is essentially asking, if you have a really big bullhorn to communicate the value that you deliver to science, society, and Earth's biota, what would you say and how would you say it? This is in the context of a hypothetical grand international event to be held next year in the Washington, D.C. area. The event can combine a conference, an expo, and potentially other things. The details are left intentionally vague so that the groups can go in a bunch of potential productive directions. However, we are asking that the groups deliver 5-minute pitches at the end of the day to address audience, motivating message or theme, delivery method, title for this program element, as well as companies that could sponsor or co-exhibit as an activity complementary to your program element. We expect solid pitches, but I'll point out again, this is a low stakes activity. We also expect that the conversations around it will be at least as valuable as the pitch. Many interesting ideas emerged from our group conversation yesterday. I'll amplify 2 of them. The first is the idea of funders as an audience. I believe that David Breen brought up the point that every US taxpayer is a funder if we're getting funding from the US National Science Foundation or other funding agencies. We need to be ready to explain the value that we bring to the world with their investment in our work at any time. You have an explainer that you share with your family at holidays. That could be a good start. I always imagine my parents as my audience. Before their retirement, they were no-nonsense small business owners. The other is an idea that Rob Gorelnik, Mike Webster, Kit Lewers, and others brought up, and that I discussed a bit at the first workshop in Florida. Collections digitization is just a step in a broader biodiversity research enterprise that has as its beginnings the biodiversity out there in the world, but now includes a bunch of additional strategies for generating relevant complementary data. This includes things like remote sensing, but also observations documented with digital media by the public using iNaturalist, eBird, and similar resources. If we view this as a positive sum game, how can our collaborations with those engaged in the complementary activities multiply the relevance of each? We have a fantastic lineup of speakers for this morning's talks. Yesterday's talks were, excuse me, been trying to keep from coughing. Yesterday's talks were billed as rapid specimen digitization, storage, management and sharing, information ecosystems, pipeline, and linking traits. Today's talks are billed as harmonizing data for research, innovation, big data, non-conventional technologies to promote research and increase visibility. After this morning's talks, we'll have 90 minutes for lunch and a self-tour of the museum. We plan to reconvene at 2:30, spend 90 minutes on pitches, then deliver the pitches at 4. Are there any questions before we get started?
Do we have teams that we're splitting into? Have we been assigned different
No, we
groups?
haven't been assigned groups, but we will split into groups at the reconvening after lunch. Any other questions? Okay, the first speaker today is David Breen, who will be speaking on image informatics for metadata extraction and verification of museum specimen images. David, the floor is yours.
Hey, let's see. Is there a clicker up there? Clicker. All right. Let's see if I can actually. Good morning.
Go Al.
Right, thank you. Yeah, good morning. I'm going to give a talk that's actually similar to one you saw yesterday. Not quite as extensive, but for fish. And I'm not to steal an idea, but I don't know if we're going to call it fish machine. But this is work that I've been doing at Drexel with my colleague Jane Greenberg, our students, and some great folks at Tulane, and which was part of the BGNN, the Biologically Guided Neural Network HDR Harnessing Data Revolution project, which has been folded into the Imageomics Institute. And what I guess we found that the people doing AI, that there's all these wonderful digitized collections, but there's a gap between the images and what needs to be fed into those AI models and the AI tools. And so part of what really needs to be done, so I'm associated with the Metadata Research Center. So Jane likes to say everything can be metadata. And so part of what, when you have these AI models, one of the things you need proper, correct, complete labeling of the data that you're going to analyze. And so we looked at, how do we take these images of these wonderful, nicely structured images of museum specimens, extract the metadata, verify the metadata, and then sort of think about, again, what is the information that we can extract? So I use the term image informatics. I've been using it for many years now, which I think it's kind of morphing into imageomics. So we need this correct and accurate metadata, again, for these downstream applications, and we focused on these on fish specimens. So what does it mean to make these images AI ready, right? So you need to be able to, like, you have thousands of images. Of course, you have to automate this process of finding, make sure that you that what the label says or the metadata says for that image is actually correct, right? So you may find that it may not contain a fish, it may contain a plant. There's incomplete data. So dealing with AI models where you're trying to train things, but you don't necessarily have consistent data, label data is also kind of a problem. But there are issues, again, of these different types of images that you're looking for. Maybe your tools may be looking for a particular structured image. You might have partial fish, damaged fish, things that are occluding, like I showed some nice images, but also there's image quality. I mean, the contrast may be terrible, it may be out of focus. So all these things we want to sort of work on to make the images I'll say AI ready. So some examples of, again, these images, these wonderful images on the left, which are sort of very easy to analyze. And then you have like pieces of, fish heads, and maybe I'll show some images where things are occluded and, they're overlapping, and it may confuse some of our algorithms. So we're not going to, make things from scratch. And so we go out and, again, we mentioned we're also using Detectron. My student decided to use Tesseract to do some OCR work. So there are tools out there, and we're just looking to how to piece together the tools for the applications that we want. And so again, using Detectron, you can train Detectron, again, to find the fish, to find the sort of the type tag, find the ruler, gives you bounding box. It gives you various information about that. So given that you can find these elements in the images, what is the metadata that we want to extract from these images? And so Detectron can tell us right away if there's a fish or how many fish there are. But we wanted to extract much more data about the orientation of the fish. Whether objects overlap. We wanted to extract image quality metrics. And so there's this whole list that we're able to, just from that, what we can get from Detectron. Is there a fish? Is there a ruler? What are the bounding boxes? We can get the scale. We have a way to read the ruler, which I'll mention. We look at the foreground and background around the image and the bounding box of the fish, so we can get a measure of contrast just around the fish. The orientation of the fish we can get, which way is it pointing, left or right? So those are the things that we can extract from those images given Detectron. But we did, find that what came out of Detectron wasn't perfect, and so we did things like improve the masking to make sure that we had we could sort of, given the initial bounding box, we could tweak the bounding box to make sure that we had, a nice tight fit. Well, let's see. There we go. We had, we had a nice tight fit. Well, that's interesting. A nice tight fit, or sometimes things got cropped and we're able to tweak the bounding box to make sure that we had sort of the correct fit. One of the things that we looked at, and again, I'll show that we also wanted to segment the fish, kind of tweak what's called the Atsu algorithm for finding thresholds. I like to work, for those of you who are imaging people, I just really dislike RGB. I like to work in HSV space, hue saturation value. We found that working in saturation space has provided us better ways to segment fish and not fish, tweak the Atsu algorithm to get a sort of a better outline. So this is my little trick for reading the ruler. And you look at all these papers and they're trying to find the hash marks and all that. We just found the 2 and the 3. We let Detectron find the 2 and the 3, and you measure the pixels between the bounding box of the 2 and the 3, and now you have pixels to inches, and then convert to centimeters. One of the things that we did, once we can read the tag, once we can identify the tag, we OCR the tag, and one of the things, for the pieces of metadata we wanted to verify was, is the name correct? So like the metadata says this is a such and such species fish. Well, is that correct? For the most part, it is. We looked at over 7,000 images. This analysis did find more inadmissible images. I'll call some things that are problematic. Fish where there seemed to have 2 names, as in the person identifying it wasn't sure exactly what kind of fish it was. There are synonyms. So we did find, yes, there were 15 out of 7,200 labels that were incorrect. One of the things we got interested in, because we're interested in kind of these looking at shape as a feature. Again, we had that improved segmentation. We find these outlines, and then sort of looking at, can we use these things called elliptical Fourier descriptors as a feature, again, for further downstream analysis. And we did our own kind of quick analysis to say, yes, that for 5 or 6 samples that we looked at, 5 or 6 type species, that this thing called the elliptical Fourier descriptors actually give you a signature you can use to identify different fish. And there's sort of our pipeline. Again, just kind of, Detectron finding the fish, using kind of our own method for segmenting, getting the mask for the fish. We rotated it because we needed, for our analysis, we needed everything in the same orientation. And then, you do this pipeline, you come down to 6 numbers, and sure enough, those 6 numbers that you can distill from this process can help you, is a signature, can be considered a unique signature for various species. So anyway, we've been publishing with our Tulane folks, got a bunch of papers on the things I just described. And of course, again, our Jane Greenberg, who's actually leading this effort at Drexel, are great students, and also Yasin is here from Tulane. We've really enjoyed working with them, and there we go. That's my lightning talk on what we've been doing for the past few years.
Hello, everyone. Yasin Bakış from Tulane University. So I'm going to talk about our experiences with the machine learning scientists and some machine learning pipelines that we have been working with. So with the BGNN Biology Guided Neural Network project and ImageOinks projects, 2 projects, we aim to do some machine learning analysis with machine learning scientists. We are biologists or biodiversity informaticians. Our aim was giving them some data set and the biological guidance, and then let them do their job. And that was pretty straightforward. I asked Hank, he said, just download the data, push them towards their way. And I said, oh yeah, that's very easy. And then those are the images that we are looking for. However, the images were like this. And we said, okay, like we cannot give them all those data sets. And this data set is coming from IDBio. If they have fish in those images, then 50% of them look like what we want. And then there are a lot of non-fish images. Once you search for like all the fish taxons, you get around 500,000 images, and at least 400,000 of them are like this. So how we are going to do this? And we ended up creating that workflow to make everything tidy. I can come back to this image in the Q&A session, but this is basically if the data is coming from this platform or this museum, then do this and clean up, filter, or like capture some metadata and then make it available on the API through API, and then gather the results and receive some feedback and those kind of things. And the first thing we did was doing the filtering based on physical properties, file size, which is pretty straightforward. According to file size, delete the lowest size images, or look at the dimensions. But they're usually good. And then we captured this manually. We used students and we hired some contractors. We capture all this data for every single museum image. And we thought this could be very helpful for the machine learning, and it was. Some of the publications we did with David and Anuj and others are based on that. And those are very fresh results. And if you stretch every single image to 0-1 distance, you get those distributions, fish on the top left, scale bar, color bar, eye, labels, and barcode. So those are some of the objects that we captured through object detection pipeline. And you see like each of those colors belong to the museums, different museums. So you see museums have a profile. So they don't place the objects randomly. They usually pick a place, and in our case, it is fish specimen. Then fish is always located somewhere, and color bar is somewhere, or like label is somewhere in general. And then we are extracting some kind of museum profile. And you see there are some mistakes on the size, for example. Then this is how we remove those mistakes, because a specimen needs to be around the center, right? You never put the specimen on the side. And then this is on the left, single fish specimen, on the right, multiple. Whenever it is multiple, that purple museum tend to, what is it, OSUM? OSUM tend to put them on top of each other as a line, and they're always on the left. Or INHS, they never do that. They never capture multiple fish specimens. So they have like those kind of dynamics. And so those are the other features that we are extracting. This is like each of those is another work, but we are adding all those data to our FishAIR. Object detection, annotation, midline extraction, scale count, closed curve, outline. Outlines are in different forms, and then histograms, color palettes, and data provenance. Data provenance is very important, because once we generate the information, then we want to be able to inherit the information, because we don't have single image. We keep producing images through our pipelines on the right. To be able to access those information is very important. And then this is David Breen and what Drexel metadata group has been working on, and they produced a lot of data that we still haven't been able to embed to FishAIR, but this is our future work. And then once we have all those data generated, metadata generated, what do we do with those? So we started with only like 5 terms in a CSV file, in a text file, and we were being as simple as we can. That was the point. And then they kept asking, hey, like, can you add this feature? Can you add this feature? So we ended up like around 120 terms in our vocabulary. We call it AI-Readiness vocabulary, shortly AIR. And only borrowed terms are like around 10, 16, the yellow boxes. And in the future, Max and Provo, 12 and 9. Out of those 120, almost 80 of them, I guess, are brand new. So we are planning to work with Steve and Tadvik so that we can make this publicly available so everyone can see, everyone can benefit from that. And we are not professionals when it comes to creating those terms or vocabularies. So you can see this in this address in FishAIR in the vocabulary tab. And then we created a metadata schema. And what we did, the most important thing here is the batch concept. So we don't take images in singular form. Instead, whenever we create images, we create them as in batches. So we keep the all the information based on the algorithm that the data has been produced, or like who produced this, when, and like publication, if there is any publication, those kind of information. And those are all the data that we have produced, around 40,000 images, and almost 300,000 of them are AI ready. And what I mean by AI ready is within your pipeline, if you have a pipeline like a landmarking pipeline on the right, from museum image to bounding box image to segmentation, and then landmarking. Now you have so many images that are produced from each other. So you need to keep the provenance information, and then you need to be able to rename all those files, and you need to send them to some kind of repository. But this needs to be very quick. So FishAIR provides you like, hey, can you give me the parental identifiers? Then I can provide you with the unique identifier for your file names. And then you can continue, and I will provide you with the data that I have, or metadata that I have. So this is very fast. And in the backend, we continue analyzing those new images so that you don't have to create bounding box again if someone has already done this part, or some people did the segmentation, so I don't have to start from the scratch from the museum image. So we keep all of those images in the FishAIR. And if there are different batches belong to the same pipeline, then there will be multiple batches. So you can pick whichever you want. And back to the our pipeline. So we downloaded all those data, we cleaned them up, we filtered them, we created some metadata so that they can filter again. And then we provided those archive files that includes all the images that they want, plus the folder hierarchy, file folder hierarchy, so that they can start their machine learning pipeline or neural network pipelines easily without no problems. We create them some scripts, and everything is good. This is my last slide. So on the left, what we have done so far. On the right, what we are currently working on, or what we are planning to do. Most important ones are AI readiness is very big because AI is getting big, and we are not ready as museums or data providers, data repositories, data creators. We are not ready for AI. So data provenance is important, and image quality is very important. And then AI fitness score, we are generating an AI fitness score for every single image, every single museum, and every single taxonomic group. We are going to produce a lot of feature extraction features from the neural network pipelines. That's all. Thank you.
All right, Anuj, you should be able to share. Meet us.
All right, I'm starting to share my screen.
You can see it. Yep, looks good, sounds good. Thank you so much. Take it away.
All right, okay. Thank you everyone for coming to this talk, and I really wanted to be there in person, but I think this virtual talk would work just fine. So I'm Anuj Karpatne. I'm an associate professor in the Department of Computer Science at Virginia Tech. And I work in this area of scientific knowledge-guided machine learning. So what I'm going to talk about for the next 10 minutes is how can we use scientific knowledge or biological knowledge in this particular project's context to develop better machine learning methods that can enable the discovery of biological traits directly from images. This talk builds upon what Yasin already presented, that we are working on this image geomics project. A precursor to that was the BGNN project that Yasin also talked about, where we are trying to see how we can use images as the source of information about the rules of life. So very quickly, let me describe the goal of the image geomics project from the perspective of knowledge-guided machine learning. So image geomics is a brand new term that we have coined, and it's supported by this NSF-funded HDR Institute grant, where we are looking at the omics of images. That's what image geomics loosely stands for. That given images of different organisms, can we arrange the information of these images in a structured way, guided by biological knowledge, that can enable us to solve a variety of tasks that biologists care about. For example, looking at all the images that we have from FishAIR, or from the other museum collections individually from IDIC-Bio, from INHS, OSUM, and other sources. Can we use all these images of fishes, butterflies, birds to, let's say, do species classification? Can we extract latent representations from these images that can enable a variety of tasks? And species classification happens to be one of those tasks. We can evaluate where current state-of-the-art foundation models are when it comes to solving biological problems like species classification. So we are doing a lot of benchmarking analysis of pre-trained vision language models. These are the kind of models that are behind ChatGPT, or recently GPT-4o and other models that you may come across in the media. We are evaluating all those models on biological data sets as part of the image geomics project. We are also developing specialized models for different biological tasks like trait segmentation and trait detection, enabled by all of the work that Yasin mentioned by curating the FishAIR dataset. But another aspect that we want to explore is that, along with identifying traits that we already know that biologists care about, for example, known landmarks, known regions of these species, can we discover novel traits, novel regions, novel landmarks that we have not focused on so far? Given that we have large scales of data sets from different museum collections, can we try to see what computers can see that we are not focusing on so far? This is the goal of discovery of novel biological traits directly from images. And to do this, we want to make use of biological knowledge available in different forms. For example, for evolutionary problems, we can look at the knowledge of the tree of life. And there are other forms of biological knowledge, for example, the phenoscape, ontology of the different anatomical parts. So let me, in the interest of time, talk about just one of those case studies that we are exploring in our group, which is on discovering evolutionary traits from images. So let's say if you are given images of 3 different species of birds, and if we apply a machine learning model that extracts features from their images for species classification, right? This is a standard task that we would see in any mainstream machine learning application. But in this particular problem, we know that these species do not have a flat structure. We can arrange these species classes in a tree-based structure guided by the tree of life, where we would know how and when 2 species diversified from the other species. We can find out what is similar at a molecular level between, let's say, the Western grebe and the horned grebe, right? That share a common ancestor. So using molecular data, given the phylogenetic tree, we know the similarity between different species. But now that we also have images of these species, we can ask the question, can we identify regions in their images that correspond to their similarity at different levels of the phylogeny? For example, if Western grebe and horned grebe share a common ancestor, right? What was the trait that they had acquired that is different from a sibling node in the phylogenetic tree that branched off to create cuckoos or other species of birds? This requires moving beyond predictive accuracy. That is one of the gold standards of mainstream machine learning. And we want to move beyond predictive accuracy to focus on explainability and scientific discovery. We want to capture the information of these images, not just so that we can classify them, but also to discover regions of their images that explains why they are different and how they diversified in the process of evolution. To enable this, one of the methods that we have developed is this idea of phylogeny-guided neural networks, or PhyloNNs, where we want to encode the information of images of different species in a sequence format where different segments of the sequence capture information at different phylogenetic levels. For example, we can have the first segment of information capturing information that is shared across all species that descended from this gray ancestor node, which would cover all these 3 leaf nodes. At the next segment, we can identify what is the sequence of features that is shared between the grebes, right? Which have the blue ancestor node that is different from, let's say, this cuckoo bird that had a different ancestor node at that particular level. And we can keep on doing this at different ancestor levels so that we have an entire hierarchy of features where every segment of the hierarchy captures information at a different point of time in the process of evolution. Once we do this, we can also try to analyze what these different segments of information look like in the image space. So more recently, we have been looking at generative models, for example, diffusion models that are behind state-of-the-art products like DALI, where given a text caption, you can generate images of cats or an entire distribution of images of cats. But we wanted to explore if we can condition these diffusion models or more generally generative models using tree-structured knowledge. For example, for every species, can we generate images using conditioning vectors or embedding vectors that is structured by phylogeny. For this, in our recent work, we have developed this framework of phylogeny-guided diffusion models, where very similar to phyloNN, we are learning embedding vectors where different segments of the vector capture information at different phylogenetic levels. Given these embedding vectors, we can condition a diffusion model to generate images of or synthetic images of known species. And we can also do some perturbation experiments. We can perturb any segment of this embedding vector to generate hypothetical or images of hypothetical species to understand what exactly is learned at every segment, right? We can visualize those changes. For example, just by changing this last level, we can observe some changes in the generated images, right? So this can help us explain what we are learning at different phylogenetic levels. And there are different types of perturbation experiments that you can do that are analogous to, for example, gene knockout experiments. Or we can do experiments similar to CRISPR style swapping experiments, where we can take a particular segment and replace that in the sequence of features. And this is helping us reveal trait differences that are still hypotheses that the biologists need to explore, right? So this is just giving us the tool so that biologists can ask questions about how these species have evolved, what are the features that they share at different phylogenetic levels, and can we visualize those features by doing these experiments. In the interest of time, maybe I'll just skip over and talk about another line of work in the same area of methods of studying macroevolution using knowledge-guided machine learning, where we are now building upon explainable models like prototypical networks. Here, the goal is that given an image of a species, we want to not just predict what species class that a specimen belongs to, but also identify what parts of the image or what prototypes of those in that image are being used to make that prediction decision. There is a whole family of works on building prototypical networks, on identifying these prototypes for every species. What we have developed in our recent work is to arrange those prototypes on the tree of life, on the phylogenetic tree, to identify what is the prototype that is shared across all these bird species in this example that are descended from the green node, that are different from all of the other birds that descended from the red node. So there are again many interesting hypotheses that we are generating by scaling these methods to large collection of images. And I'm happy to talk about this in more detail in the Q&A. But this is just one of the examples of what KGML can bring to power by looking at the scale of images that we have from IIIF Bio and other museum collections, and by leveraging recent advances in machine learning and especially knowledge-guided machine learning. So with that, I'd like to conclude by thanking all the collaborators who are behind this work and the students who are the real folks who are driving this research. Thank you.
Oh, okay.
There you go. Now they can hear you.
Great. Yeah, so here we're going to talk about an ongoing project about extracting phenological information from images and building a global database, which we call Phenobase. And first, can I get this to work? All right. While plant phenology, it is the timing of the key kind of a life history events, for example, the leaf out, timing, and flowering or fruiting, right? So, but why plant phenology? I think it's critical to study phenology because they are going to have, important consequences to both ecosystems and our society. For example, the timing of leaf out is going to affect the productivity as well as the length of the growing season. And down the road, it's going to affect the carbon sequestration as well as feedback with the climate, right? And the timing of flower, for example, can affect both ecosystems like pollinators, their fitness of the plant itself, as well as human health, right? So if the timing of the pollen season moves earlier, then we're going to be expecting some kind of pollen allergy season also we need to getting propelled. And fruit timing will can also affect lots of agricultural crop yield as well as some other economically important fruits. So they all have important consequences. And even more important than I think, phenology is probably one of the traits that are the most sensitive to the environmental change, right? Like with warmer temperature, we're going to see early spring, right? So given all this together, so it is very important to look at the plant phenology, and which of course all starts with getting phenological data. So where we are in terms of our effort to put together plant phenology data, I think currently we have two types of major data types. So the first is the human observations. We can have in situ observation as well as potentially getting this kind of information from images. And the second type is remote sensing data. And then we have, a different way of going to the quantity control as well as publishing the data with metadata. And here, I'm just mostly talking about human observation data. So over the past years of working with collaborators on projects about plant phenology, we realized that there are some key limitations about the current in situ or observatory data of plant phenology. The first one is the incompatible, of the existing data. So in US, we have this US National Phenological Network as well as NEON. And in Europe, we have this pan-European network, and all the country or regions, we also have their kind of, network to monitoring and document plant phenology. But there's a key issue here, like even you can get a copy of all this data, it's not easy to just grab them and use them because you use different terminology to describe phenology. So you need to put a decent amount of effort to try to integrate them first. So that's the first key limitation. And second, we have like a huge spatial as well as taxonomic gaps of the current phenology data. And here is the kind of a map of existing open access phenological data, the record of the observatory data. And we can see most just like US and Europe, and the rest of the world, pretty much like, there's no open access phenological data. So that's a huge spatial gap. And not surprisingly, just like, other traits, we have this like a typical log ratio distribution of the record of the plant phenology. Like most of the record belong to a small number of species. So we do have like a huge taxonomic gap here. And finally, we have a lack of historical data. We probably have some data, back to the 1800s, probably. I mean, the longest one we can think is probably the Japan, the cherry, right, blooming, and we have over 1,000 years of data. But I mean, that's probably just one or two species. And the majority of data took off since 2009. So before then, we have a huge gap. And I think this is the gap we're going to talk about here mostly. And I think, IDBio or digitized specimen in general can provide this essential information about historical data of plant phenology. And here is a temporal coverage of IDBio digitized specimen. And I think, if we can use some kind of a machine learning approach to extract this information, that would be great. So then the question is, can we actually train the machine to do this? And if successful, this is going to be a, enormous saving of human work, and also going to provide this like really needed historical data about phenology so that we can understand how phenology have changed over the past decades, right? With the ongoing climate change and all the environmental change. So our kind of initial testing on some of the common general suggests like, deep learning approach can be very successful. And here we can see the machine learning accuracy is actually pretty good. So blue means like higher accuracy, and some of general we have higher accuracy than human kind of expert annotation accuracy. But the big question is, can we actually scale this up, right? Not just like tens of thousands of images on some general, like that's pretty common and easy to detect, but across, like scale it up to all angiosperms across like 50 million images, right? So that's the big question. And then here, just like this is an ongoing project, but we are still kind of in reasonably early stage. So here are some kind of the key challenges we think we're going to need to overcome. And the first is to actually get access to the specimen, digitized specimen images. IDBio provides the metadata, but they don't host a copy of the images, and we actually need to go to different portal to download all the images. And after then, think about how can we assemble good training data and potentially take advantage of the recent development in AI methods. And also, once we are be able to apply the models, and we need to carefully validating the results, and after then disseminate the data. So I think yesterday we talked about how to lower the barrier, right, to the users, so they can use the data. And I think that is critical, if we wanted to make this useful. And later, and we also talk about a futurism, right? Like in the future, with more coming digitized specimens or iNaturalist images, and then how can we actually keep the data sets dynamically updated with new image coming, and we can digest them frequently. And then ultimately, as any project, we need to think about, what is the sustainability plan here. So putting all this together, so again, we're just trying to build this global database of phenology, which we call Phenobase, by integrating existing in situ data and extracting new phenological data from community science or citizen science images, as well as digitized specimens, and putting this together. I think Phenobase has a huge potential in terms of research as well as education, because phenology is kind of an ideal topic to get involved with the general public or students, and that's another topic we talked about yesterday. And with that being said, we are actually still looking for a postdoc or data scientist to work with us on this project. So if you know anyone, or just spread the word, that would be great. Well, that's all. Thank you.
Right, so we have a little time for questions. If you are in virtual land, you can put your question in the Q&A. If you're here, you can raise your hand. Anyone have any questions for our first round of speakers? Kit.
I have a question for the presentation that just happened. Have you thought about using like a web scraper, like just like HTML based to get the images? Because like NASA does the same thing where you have to click into each individual image to like download it.
Uh-huh.
And I found that like, I was like trying to overcomplicate and be like, I need access to your API and all these things. And I just
Yeah.
made a web scraper and was able to get all of those images.
That's what we're doing with INET at this moment, because INET has this Amazon open data repository, so we can actually just get the photo ID and paste the URL and get it. But the specimens, some of the portals, they have this dynamic, database.
Lovely.
It's just pretty hard to standardize, I guess. Rob, have you thought about that?
Yeah, we kind of do have effectively a web scraper that tries to collect the images. It's just a little tricky at the edges because of fail rates on some of the image downloading steps. And it turns out like this is kind of no complaint exactly because IDBio is a lot to manage, but like trying to like get all the angiosperms downloaded turns out to be like more of a challenge than you might first think, because like there's no completely standardized taxonomy that allows you to traverse networks as easily as you want to. So like just deciding what images actually fit our kind of taxonomic scope and then downloading them turns out to be like more tricky than we had hoped. We've kind of solved those problems for the most part, but I think we're still missing. I mean, it's no big deal. Like, we missed 50,000 images out of 30 million. Big deal maybe, but like it's still a little annoying that we can't quite get there.
So I'm just curious, like, do you have a position on like licensing of images and your use of them? Like, do you consider it to be data and therefore the license is irrelevant, or how do you understand that issue?
Is that for us or for?
I don't know, anybody, all the people who are getting images and.
I'll answer for us real quickly on our national list. We worked with them to decide that we're not operating and creating any annotations on anything but CC BY or CC images. So any image that's owned by, that has a copyright that's owned by the original provider that isn't an open license, we're not gonna touch.
So, but I'm assuming like the attribution part, you just for like. You're not recording.
All the records, all our annotations are gonna, without fail, point back to the original records. And we argue that's sufficient for us to make a case that we're following attribution.
Cool.
There's also some debate whether the metadata or extracted information from image is copyrighted.
Right.
So in our case, every data that we provide, whatever they say, we get that information, licensing information in the metadata, and whatever we produce as CPBY. But I'm thinking after like yesterday's discussion, if we have like funding problems, then we might go for like more about, because we need some funding, we need those organizations or companies our process. We need them to support our work. So we might have to think about that a little bit.
I would take care in how you think that through and tier it. I can see why you should have companies cover costs, but for academic users, I think you're gonna limit the audience.
Well, if they need the data, then they are going to use it anyway. So the problem is a lot of data available right now out there, and even our personal data is available to companies, and we don't get anything. But this is about work, and we are producing your data, spending some money, and if they are using it, then they have to contribute to that study. And if
there's a commercial aspect, or?
If there's commercial, definitely, but even if it is not commercial, we might. So if I am using all those museums data to create something, then I am getting some funding from NSF. Then I should ask for, okay, like I want to support all those data providers. We should have that kind of thing, even though those are freely available.
We also have a question in the chat from Nikki Nicholson. It says, for images derived from specimens, has there been any conversation about the opinions of the countries in which the specimens were originally collected? Perhaps mirrors the conversations about sequencing.
Can we read that again?
Yes. For images derived from specimens, has there been any conversation about the opinions of the countries in which the specimens were originally collected?
The countries?
Yes.
Well, in our case, they're all coming from US.
I know at the Smithsonian, I can't speak for them, but there's some countries where there are no images put online for those specimens, even though they're taken. The botanical specimens may be.
I have a question for Anuj on Zoom. Great talk, Anuj. That was very interesting. I'm wondering if you've thought about applications in paleontology, for example, to look at admixture between different lineages and how that affects the traits. So if we have a paleontological sample, can we apply these? And rather than think of it as CRISPR experiments, try to work thinking about admixture.
Yeah, that's a great question. Actually, we haven't looked into paleontology yet. We have only been looking at images of species that live today. But it would be great to explore paleontology images. What I understand is that the modality of those images may be slightly different because they may not look like the fossil records may be of varying natures. For example, it may just be an imprint in the soil, or based on my understanding, we would have to think of how to use the larger collection of images of living species and still use those niche modalities or those modalities of images of fossils records and paleontology records where we have lesser number of images. But certainly this is very valuable information that we haven't used yet. I'll be happy to explore that.
Yeah, thank you.
I had a similar question and follow-up, whether it's biological or paleontological, do these methods work for quantitative analyses, or is it just presence absence of trees?
So we are doing some. So did you ask about qualitative analysis or quantitative?
Quantitative, so measurements rather than presence absence of traits.
Right, certainly. So we are working on measurements of traits as well. For example, once we have the segments of the different fins of fish, we can calculate the areas of those fins. We can look at the distances, and Yasin has an entire pipeline of barcoding these fish specimens once we generate the segmentations. So those segmentations can be used for presence absence detection of traits, but we can go beyond that because we have the pixel level annotation to talk about shapes, areas, angles, distances, and other such metrics.
I had a quick follow-up on that. Sorry, Anuj. If you give it the wrong tree, can you force like, fake homologies, if that happens all the time, right? If we get get multiple species trees and we're trying to work out what are the traits that unite these things. Have you tried that?
Yeah, that's an interesting question. We haven't explored that. So there are two lines of work at a very high level. One is where we provide information about the traits that we care about, and we want machine learning to find those traits. For example, we know these 7 types of fins and we want to find them. The second kind of work is where we provide no information about traits and their morphology. We just provide images and we provide some structure, for example, phylogeny, and we ask the model to find interesting features by itself. And it may happen that it finds features that are not as interesting because of synapomorphies and other convergent processes of evolution. There may be some traits that a model discovers from images that that is not as interesting. But yeah, we haven't explored like fooling the machine learning model by providing wrong traits.
Okay, thank you so much to our first round of speakers. Julie Winchester is up next again to talk about IIIF this time. Thank you so much.
Yeah, hello again. Yeah, so yesterday I talked about my work with the 3D data repository MorphoSource. Today I'm gonna switch hats a little bit to talk about to give a brief introduction to IIIF, which is another organization model that I work with as a specification editor, and also as one of the co-chairs of their 3D technical specification group. Though I'm mostly going to talk about 2D. I'll talk a little bit about 3D. And we just saw some really amazing, honestly kind of mind-blowing like imageomics talks. I'm going to take a step up conceptually, which means I'm mainly going to show you some pretty pictures, and it's not as intellectually deep. But if you imagine, you know, like the flow of data coming from providers and flowing downhill, we're going to take a couple steps upstream to talk about what's the best way to structure that flow. We're going to get back into this data ecosystem stuff. But to start with, what is IIIF? So that's a mouthful. It stands for International Image Interoperability Framework, or as we IIIF. There you go. But that's okay, you just threw a bunch of words at me. What does that mean? IIIF is two things, really. It is a standardized model for using the web and other technologies to present and annotate content such as images and sound and video, and soon 3D with lots of other benefits. But more importantly than it really just being a model, it's a global community. It's a large, active global community that develops open APIs, implements those in software, especially a lot of open source tools, and groups that expose images and video and sound and soon 3D in an interoperable way that allows for communicating sometimes complex digital objects. Just to sort of show, there's a decent number of people working on this. This is from 2019 in Göttingen. I think we finally have a better picture of people from the conference that just happened in LA. I just don't have that picture yet. This is a map that shows just institutions that are using IIIF right now. And just some names of some large leaders. I mainly threw this up here. Yale, our host, they are among the international leaders. You may also be seeing your own institutions there. And what are the contexts where IIIF is used? Pretty much anywhere that images can be used in an academic sense. We're talking about libraries, museums, scholarship and research, teaching, digital humanities, AI and machine learning projects, like a lot of we just talked about, but also STEM in bioimaging and life sciences. And at the end of this talk, I am going to back around to talk about IIIF with biodiversity. But before I do that, I just sort of want to take you through some of the things you can do with IIIF, which normally this is the slide to talk about why images are important, but I feel like this is a group, even digitally, that does not need that. You have had a whole hour of the amazing things we can do with images, how they transmit knowledge. So instead, I'm going to show you some use cases of what IIIF allows people to do with images in a very interoperable, standardized way. So let's just start with a simple use case. Let's start with the problem first. So this is sort of reminiscent. Rob showed yesterday the silo diagram. Obviously the same thing with images. It's just the equivalent that if we all have our own infrastructures, it has limitations. One of the things IIIF allows you to do is to move across this, to break out of the silos, to expose data in a way that everything can get at it. Okay, yeah. Maybe I have a very large image. So this is an image of a Japanese tax map from 1837. You can see at real person scale, this image is 11 feet by 17 feet tall. It's intended that you stand in the middle of it and sort of see it in the round, which we're not going to do. It's archival material. But IIIF allows you, so this image file is like 1.27 gigabytes. IIIF allows you to put this in a web viewer and then to do easy tiled deep zoom. So you can zoom right down to the bottom of that. Other things IIIF allows you to do, very common use case, to compare images. So these are 4 images of sort of the same street spot, which if I zoom in, you can sort of see the difference in the billboards over time. IIIF allows you to compare images side by side. It also allows you to compare one image, maybe imaged using multiple spectra. So we had some talk about multispectral images yesterday. A very common thing is that IIIF enables curtain viewers, so you can view these sorts of things. So think from the Leiden. IIIF also lets you view things that come from different places. So this is an example of a medieval manuscript where all of the illustrations, the miniatures, were cut out. They were cut out and went somewhere else. And then they eventually the pages and the miniatures filtered back into academic collections in Paris. It's great, right? Except there's 2 different collections. That's like the miniatures are at one, the text pages are at another. And while that's close in walking distance, they're different institutions. They might as well be different worlds institutionally, except that they make all this content available in IIIF. So a group through Biblissima has been able to put the pages back together, and you can actually put the miniatures back in the page. So to put things back together, to appreciate it in its original context. You can also do annotations, so people with your images with bounding boxes, both polygonal and non-rectangular, you can express that in IIIF. So this is a high-resolution image of a cell with these commenting annotations, and because it makes use of the deep zoom, students can appreciate the cell at different scales. Annotation opens up a lot of doors. Some groups use it. The Library of Wales used it to allow people to crowdsource people identifications. So, potential citizen science applications there. People are doing, I have no idea if this will work. People are doing some really bespoke storytelling with images through IIIF. People use it for maps. You can annotate. This is a historical map annotated on a modern map. And you can do this with a whole bunch of maps and see how they line up. And you can do more than images. I'm not going to show this in detail, but it works with sound. It works with video. And by June 2025, we are hoping to have a draft of the IIIF specs that will include 3D. This is very simple. It's sort of just taking this little astronaut model almost as a toy and sort of making scenes. We're going to take one astronaut, we're going to take another, we're going to make it big. But to do all the things you could just do, but also with 3D. And I think we've made really solid progress on that so far. And you could do it all in the application of your choice. So this Indigenous Digital Archive repository site uses 2 viewers. They use you can view the same content in Universal Viewer, which does deep zoom a little bit better, or Mirador, which lets you do sort of 2 images up for comparison. All right, so APIs enable reuse. But the real value of IIIF is this. This is the breaking through the silos, is when everything's available in a standardized way, and you can start to build annotation and transcription tools that pull from multiple sources, start to do cross-repository research, all of this stuff. Now, just have a few more minutes. I just want to tell you a little bit about how IIIF works. So it's made up of 6 core APIs. The 2 big ones are the image API, which is just a standardized way to get pixels from the internet, and then a presentation API that gives you just enough metadata to drive a remote viewing experience. And I'll show you what I mean by that. So in this view, the blue square, that's an image that's being pulled from the image API. When you see the sequence of images that you could click between, that's the presentation. The presentation API sort of the one API to rule them all. It's what you use to take your video, your audio, your images, soon 3D, and then to make a digital complex object that has like a viewing experience. A more complex example, all of the images you see here, the main image, some of the previews, the preview over there, those are pulled with Image API. Those are your getting pixels. Everything else, the sequence of images that tells you this is the front. We can also click over and look at the back or the figure on the left's face, the description or the metadata, all of that comes from Prezi, from the presentation API. There are other ones, I can talk about these. There are lots of other things you can do as well. All right, real quick, what about biodiversity? So there's a lot of stuff out there for biodiversity. I'd kind of like to honestly like make a list because I was not able to find a normative list of all the people that are making things. But so there was a pretty cool project through DISCO that in 2019, partially involving the Royal Botanic Garden of Edinburgh, where they wanted to bring IIIF natural history collections. Some of the products of this, so this is a GBIF record for a Royal Botanic Garden of Edinburgh herbarium specimen. They are now exposing their things. And in 2023, GBIF actually added a feature that if you have a IIIF manifest in what you are exporting to GBIF, they will give you a little IIIF icon, and they actually host a IIIF viewer. So if this was a massive image, if you did deep zoom in this, you could get all of that. And it's, GBIF's not serving the image, it's just pointing to the institutional image store. A lot of institutions have IIIF in the background. It's possible that your institution, if you're like, do we use IIIF? It's possible that your collection images may make use of IIIF. I think the Smithsonian's pretty good about it. So they have some cool things like Edward Muybridge cyanotypes. So talk about animal locomotion, some of the earliest things. But also I was just randomly clicking around one day and they had this page for kids that was like, or really for the general public for education, that was like, compare two fossils. And I was like, oh, hey, I'm pretty sure that those are using IIIF viewers. So I followed this Archimedes specimen back to their collection database. And indeed, all of that, because this is Mirador, this is a IIIF viewer. So all of their images are being exposed like that, which means they would be amenable to annotations or combined presentations or being combined with videos or audios or things like that. And said I was mostly taking off my MorphoSource hat. I would be remiss not to mention that all of our assets, even our 3D ones, are actually backed by IIIF manifests as well, which is part of what enables being able to embed that content in other websites. But soon, you will be able to take those 3D things and put them in an immersive, complex 3D scene, where I can take the thylacine and put it in a scene, like an actual, environment. All right, I like to leave on this very common IIIF, thousands of adopters, scores of applications, 1 billion digital objects, and revolutionary promise. And I will leave you on the term promise because I think there's a lot of images that have been pushed out with IIIF for biodiversity. We could probably get more collections using it. But I think that higher level, the bringing things together, the mobilizing annotations, that's something, maybe people are already doing this and I just didn't put the right slides in which case, please tell me and I will put them in for the next time. But I think there's a lot of promise for this with biodiversity. All right. That's it. Yeah. That's
All right, thank you all very much for being here. So this should be a fairly straightforward, simple talk. And you can think of it as a warm-up talk to Morris's talk that follows it. So just briefly talk about some high-level thinking that I've been doing over the last few years. And I think it intersects a lot of concepts and discussions we've been having here. So as you all know, and I don't need to spend almost any time of it, natural history collections are really the physical basis for our understanding of the natural world. And there's some discussion that's been happening since yesterday that perhaps they can be more than that. We don't necessarily need the tie to physical objects, but essentially traditionally that's the role, right? So, and because there's this kind of physical basis for how we understand the world, the concept of FAIR has always been a core component of the philosophy behind those natural history collections. And you've seen that plenty yesterday. Ed talk with an interoperability, Julie's talk just right now. Like the idea of FAIR, so make it findable, accessible, has always been a component of how we do our science. And really, over the last 20 years, we did a lot of work along those lines. So there's been a lot of digitization efforts over the last 20 years. And you have the emergence of data aggregators that not only make that data available, but make it easy to search, easy to find, very queryable, very nice. And as it was discussed yesterday, there were times, such as during the pandemic, where having those was essential for us to keep doing our work. But there's also challenges, right? Because in many ways, these portals are very, their functionality has limitations. And we've been working with systems that have been developed very much in the kind of keyword framework, right? Like you need to have a tag associated with the item for you to be able to find it. And that's kind of a large limitation because we're both users and consumers of this data. We're producers of that data. And there are always trade-offs. So you can, for example, upload your data and add rich metadata, but that takes time. And that often runs into constraints about personnel and all of that. So it's not very hard to get to these databases, find situations such as like this first one, where we have a shrimp specimen that has a beautiful image, like it's a high resolution image of that specimen. You can look at it, it's like amazing details, but that has literally almost no metadata associated with it. And the identification is like SP2 and it's identified at the infraorder level, and that's it, right? So we have a lot of objects from natural history collections that are hosted digitally, but that we know almost nothing about. They're there, but we can't find them, right? Unless you're just randomly browsing images on the web portal. And you have situations like this as well, where you have a lot of metadata for a specimen and it's identified at the species level, but when you actually go to look at the digital assets associated with the specimen, it's just an image of a slide, right? And so you may think that you would have that information, but when you actually look at the image itself, and we can visually understand those images, but not when you're querying it, right? You see that you're not your specimen, it's just a slide that contains your specimen. So this nowadays has become really a bottleneck of how we find information, especially now that we have bigger and bigger data sets, and it's becoming more and more costly to kind of keep that metadata flow going together with our assets, right? So not surprisingly, I think this has been a theme throughout the talks here. There's been a lot of interest in AI. Like, can AI provide this kind of extra layer that kind of binds these large digital collections, such as IDBio and GBIF, to the users, but that adds a lot of that functionality that we like to have, that makes, again, all these natural history collection products more fair, more findable, and interoperable. And that's something I'm really keen on developing, but it's also challenging, right? Because the machine learning community works with data sets that are very different from biological data sets. So just for example, in the context of image classification, the typical data set for image classification in computer science is like this flat hierarchies with like even distribution of images across classes. But that's not how biological data sets are organized. We have all these kind of weird hierarchical structures to our data. We have all kinds of taxonomic biases. For some species, we know a lot and we have a lot of information. But for the vast majority of species, we have very little to no information at all. And we have geographical biases in reporting. So we know that most of our occurrence data, for example, in GBIF comes from the global north, but we know that that's not where the biodiverse regions of the world are. And I think one of the most underappreciated challenges of biological data set is that there is all kinds of intra-class and inter-class conflicts that are not present to the same scale on computer science data sets. So just to give an example of what I mean by that. So you see in those upper right corner, there are two images of butterflies, and those images are actually from the same specimen. So they look completely different, but they're the same individual. And that generates a major issue because it's like an intra-class observation, but substantially from the point of view of the computer vision features, they look completely different. And we have the opposite as well. We have inter-class conflicts. So you see here these two, on the phylogeny, these two mimic species of butterflies that look exactly the same, but that belong to two different groups. So all of this makes it so that biological data sets, especially in the context of image classification, are extremely hard to deal with. And I don't think we have found good solutions on how to incorporate a lot of this. There's a lot of thinking in many of these components. So I've been really interested in kind of prototyping what that infrastructure will look like. And like Rob said, it all boils down to people. And so a lot of what we're doing in recent years is partnering people at the McGuire Center for Lepidoptera and IDIG-Bio to kind of perhaps use Lepidoptera as a study system in which prototype a lot of these tools and see how far we can get to create that kind of AI layer in between our large digital collections and the kind of research we want to do. So our approach so far has been fairly simple and is centered on a core philosophy that if we want to create that layer, one of the things we're going to have to do is breach modalities, right? So a multimodal AI, I think, emerges as like a core component that's going to be necessary in terms of integrating these kind of natural languages or more structured data sets to visual components that a lot of these models understand. And a lot of what we've been spending time recently is essentially on creating multimodal retrieval systems. So where it can actually find data using natural language or find data using image data. So the idea is quite simple. What we do, we go to IDIG-Bio, batch download all their data. And then we have a pipeline that essentially finds butterflies in those images, extract the segmented masks. And nowadays, this is fairly simple to do with like models that you don't even have to train on your data set. And once we have a clean image of the specimen, we encode this image in a vision transformer, and that essentially generates an embedding vector. So this is like, may sound like a lot of terminology, but essentially these neural networks, they learn to represent data on smaller subspaces. And that's essentially what we're trying to do. Calculate a numeric vector that represents the image data into the space of the neural network. And the reason why we want to do that is because once you have a vector database, you can query it very efficiently, very fast. You can query million record databases in like milliseconds. So it makes it very responsive. And again, just like I said, the reason why we do that is because once you have these kind of database or collection of images and we have encoded in this network, then it becomes really easy to query. Let's say that we have a specimen that we have an image for, and we don't know what the specimen is. We don't know anything about the biology of these butterflies. You can, again, encode that image in the same encoder, calculate a vector, and do a simple similarity search in this large database, find the specimens that look like your query. And you can also do the same with text encoders nowadays, because there are models like the CLIP model that breach natural language and connect them to visual concepts. You can have a natural language query where you say, like, I want a butterfly whose wings are darker brown, the outer edges to a lighter brown and cream near the body. And again, this gets encoded in an embedding vector, and you can search a database for nearest neighbors to your query. And that can look, so this is just a mock-up that kind of demonstrates that thing. And basically here, what you see is here on the left is the query image. And as you change the query image, you can kind of see how the query in the database and the outputs of the top 20 most similar images to your query look like in practice. So you see that these models become really good at finding things that look very similar. And essentially, a lot of the initial work that we do in biodiversity is that, right? We want to find similar things to the things we're searching. And again, you don't need to have clean images for that. You could do with images from like iNaturalist or whatever source you want. It doesn't need to be clean. And you can still find sometimes even like partial images where you just see one side of the specimen, and you can still find the specimens in a large database of images. And finally, you can do with language, and these models become very easy to understand things like color. For example, you want to find red butterflies. You can just type that you want to find red butterflies, and it returns a list of the top 20. Iridescent, something that people were very curious. It would be really hard with a database the way they are now to find which butterfly species are iridescent. But with a natural language query, this becomes kind of fairly similar. You can find butterflies that kind of are called owl butterflies very easily. You can find patterns like this fritillary pattern is very common among butterfly species, zebra-like patterns. So these models allow for a lot of functionality that nowadays we're very limited for. And essentially, and I'm going to end on that note, is that I think the reason why I'm so interested in developing this AI infrastructure is because it contributes to their goal of natural history collections of being fair. And I think there's a lot of exciting opportunities, but that we're going to have to figure out exactly how to approach. But I think we'll be able to greatly increase kind of the functionality that current systems have in terms of querying that data. All right. Thanks so much for your time.
Okay, hello everyone. My name is not Whoops, but there we are. My name is Moritz. I'm a postdoc with Arthur at the Florida Museum, and yeah, thanks Arthur for introducing some of the things I'm going to talk about. So my talk is more from a user's perspective with an empirical goal, which is understanding phenotypic diversity at large scale. So when we talk about biodiversity, we most often think of taxonomic diversity, so the number of species or groups, species richness, things like that. However, within a group, like for example, dogs, there can be tremendous phenotypic variation and phenotypic diversity. Which is, for example, as Arthur has shown, two sides of a butterfly can look very different. And this phenotypic, so this is also referred to as phenotypic disparity, phenotypic variation within a species or group. And across the tree of life, both types of diversity, species diversity and phenotypic disparity are not exactly equally distributed. So there are some species which follow the expectation of increasing intergroup variation with increasing taxonomic diversity. But then there are some groups which diverge from that expectation and have either less than expected disparity or more than expected disparity. And so this is an open question why life has evolved this way. And one way of addressing this is through these high-dimensional phenotypic spaces where we map out different aspects of the phenotype in a high-dimensional space, or also called morphospace, as for example then here on different groups of plants. And we can use the morphospace concept to understand why, first of all, to map out why some parts of the morphospace are more filled out or more clustered than others. And this leads to all sorts of interesting questions where we address why parts of the phenotypic space are so densely clustered and other parts are not. However, to do this, we need really large amounts of phenotypic data, and we don't want just unidimensional or low resolution data, but ideally we want really highly resolved phenotypic information from a lot of individuals. So we both want the extensive part, large amounts of specimens, and the intensive part, we want to sample each specimen intensively. And that's, of course, that has been a big challenge traditionally. But now, I don't think I have to introduce this too much to this crowd, that natural history collections are, of course, an amazing solution to the first challenge. So they contain large amounts of very standardized specimens, and we're already making good progress on digitizing most of them. So this is really good for the extensive part. On the intensive side of things, there have been a lot of advances in, for example, computer vision, especially with the recent developments of AI that really allow us to collect large amounts of really all sorts of interesting information from a large amount of specimen. And so really for the first time, we're able to address these questions in a phenomics approach. To, for example, ask questions about diversity disparity relationships, such as how on really large taxonomic scales does taxonomic diversity relate to phenotypic disparity? How are taxa, different groups of organisms, distributed in phenotypic space, and which processes have been driving phenotypic diversification and phenotypic evolution? And I'm studying this with a large group of butterflies, so nymphalid butterflies, and yeah, as Arthur already introduced, we rely heavily on online databases for our research. And to just give you an overview of what we're doing, so we've downloaded, as of now, we've downloaded approximately 350,000 images from both IDIC Bio and the GLIF database. And then we use an automated pipeline, an automated segmentation approach to extract the specimen from the image. We use image embeddings to cluster them and use that to both filter and clean the data set on the one hand, and on the other hand, we also use these embedding vectors to address our questions and map the specimens out into phenotypic space. And yeah, I won't be going too much into detail in the interest of time, but I just want to say that, for example, this part, the cleaning and filtering is a lot of work, and this morning we've already seen some different approaches, so it is a big challenge, and I'll just gloss over a lot of things here. Yeah, that we've done, but I'm happy to talk more about this later during the Q&A. So for image segmentation, we use Meta's segment anything model that has already popped up a few times, and it's really good at detecting things in images without any prior information on that. So really powerful. We actually use a variant of segment anything, which combines the grounding Dino framework, which has enhanced text handling capabilities, so you can give it potentially more complex textual descriptions. It has just a better semantic understanding of the text you give it, and so we use this grounded SAM and we apply it directly on our data set, and it's really out of the box already giving us what we want. And it's also, so these are actual segmentation masks, and yeah, it's also really good at finding partial images. So this is good and encouraging on the one hand, that it really you know, if you show this to someone like, what is this? But Sam or grounded Sam accurately identifies it as a piece of a butterfly. But of course, this downstream also creates new problems. What do you want to do with a bunch of like partial butterflies or like wingtips? So, so this involves a lot of filtering. And as I mentioned, we do this based on image embeddings that we extract using image encoders. So what is that? An image encoder is actually just a pre-trained classification model. So in our case, it's a pre-trained vision transformer where we supply just the segmentation mask, so the foreground, and then the image encoder spits out an image embedding. And this is, as Arthur already mentioned, just a condensed vector representation of visual content. And this is really anything that the model associates with the image you give it. So this can be color, pattern, shape, smaller spatial or larger spatial relationships. And yeah, so this is all of these vectors then in the end have a fixed length and can be used, for example, in clustering. And so, for example, when all of these partial butterflies cluster together in multivariate space, we can give it a label. So we use supervised learning to classify this as junk and then remove it later. But we can also use unsupervised clustering, where we take the remaining embedding vectors and project them into phenotypic space and see, for example, the number of clusters we get, so the number of groups or different species, and use this to answer our questions. And this is an example of what we get. So this is a multivariate. This is a multivariate space created from a t-SNE visualization. It's just a technique to cluster and visualize high-dimensional data, and it groups it groups observations that are more similar to each other, closer in space, and objects that are specimens that are more different from each other further away. And we can zoom in on some examples. So this already hints at the fact. So this big cluster here in the center, where lots of the different colors are genera. So to just make it a bit more visually comprehensible, we didn't, for this example, visualize it by species, but by the genus level. We can already see that in the center, where this big mess is, there are lots of really similar species that overlap. So this already is a first hint at the fact that taxonomic diversity and phenotypic disparity are, at least in this representation, not equally distributed or aligned across this space. But there's also some other interesting patterns. So this is this is a genus of fritillary butterflies. The fritillary means dice box. So that's because of the checkered pattern. And this particular genus essentially consists of one species. And we see, except from this middle part, we see 4 clusters. So what is that? This is simply female ventral side, female dorsal side, male dorsal side, male ventral side. So the fact that using these embeddings, we can so reliably separate species within or clusters within a species, allows us to answer all sorts of, or address all sorts of interesting questions, such as how did dorsoventral dimorphism evolve? How did polymorphism evolve? And so this is really promising. The next step is to integrate phylogenetic information. So use one of the most recent phylogenies, and then just take it from there. So this is very preliminary. And I'm at the end of my talk. Thank you. I thank my collaborators and Arthur, and you all for watching.
Yay.
You. We have about 7 minutes for questions.
I have a question for Julie and this IIIF. I mean, I know the graphics community has been looking at standards for presenting 3D models on the web for decades, you know, I don't know, starting with VRML, for example. So how is like what you're doing, getting into that, building on it? I mean, just wondering how this ties into in the web 3D and all the other activities standardization activities that happen in that area.
Yeah, no, that's a great question. Yeah, thank you for that. And I will say I could have given a whole separate 15-minute talk on the 3D work. Actually, I just gave one last week. So if anyone, I guess I could share that if people are really interested. But yeah, so actually speaking of VRML, because yeah, if people don't know, yeah, VRML became one of the few like ISO standards for 3D on the web, and then it evolved into something called X3D. And we actually have one of the people that works with the X3D group is also in this technical specification group for IIIF. But what we are targeting for this is something similar to what IIIF's presentation API enables for 2D, where its presentation API is not trying to duplicate Photoshop, and it's also not necessarily trying to duplicate every possible thing you could do with images. It's trying to give you a good way on the web to do sometimes, you know, presentations that involve complex assemblages of things. And with 3D, we're targeting a similar level of abstraction. I think a thing we like to say all the time is we're not creating a new scene format. Because, you know, like another big format, because like X3D can be like a full scene graph, or like even a file format like GLTF can be a full scene graph. We are targeting a level of abstraction a little higher than that. You know, you're going to be able to put multiple models in a space. You will be able to add cameras and lights to that space. You'll be able to combine 2D and videos. You'll be able to place, you know, like a 2D image in the space. And you'll be able to have some, you know, storytelling in terms of moving things from position to position. But it doesn't try and get, it's for like the broadest common denominator presentation tasks. We're not getting into the scene graph level or doing like deep animation, or like it's not a physics engine or things like that. Does that go? Yeah, we're trying to hit slightly above on abstraction compared to a lot of those like web scene graph stuff.
Rob?
Actually, I think Nicola was.
Go ahead.
So I had a question for both Arthur and Moritz. So I think the power of the embeddings is great. I mean, we've used it before. So I want to just either of you to comment on how you pick the metrics. And then when you're using TSNE, it's a great viz tool and it shows you that the clustering works, but it's not a clustering tool. And so have you addressed like how you would now go, and or are you just using whatever metric you're using to do clustering? Because like, you know, if you have new data coming in, you can't use whatever TSNE did to cluster that new data. You have to start all over again. And so how are you handling that clustering with new information coming in?
I don't know. Well, either way. So one of the things we do with regards to cluster is that we do on the full multivariate vector. So actually like I said, TSNE is very useful for visualization. But if you have a multivariate vector, you could just do clustering, and that's good.
But how did you pick your metric on the embedding vectors? Like how?
So these models have all been trained on metric learning. So they essentially lie, what you're gonna call on a hypersphere of any dimensional hypersphere, and you can calculate distances between things within that sphere, and so then those distances have meaning. So that's essentially how we train those.
If maybe I can add that this was really just a first pass on what is going on, how is our data situated, and we really, so for the trash removal, it's really quite useful because you just see a big cluster of junk detections that are just look different from a butterfly, and then you can just, using an interactive tool, you can just select them all, and then you can do anything, like you can pass this on to another classification step and then remove this. It's really just a first
yeah, no, I think it's great. I just was, you know, wanted to hear more about the next steps.
Yeah.
So.
Hey, Moritz and maybe Arthur, I love the work. I think it's super exciting, and I think there's lots of potential. But I have a biological question for you guys. Like, so you know, you're using biased taxonomically and spatially and temporally biased data in. So how do you deal with the fact that, like, if you're just downloading the images off IDBio that have been digitized, you're not really capturing like butterfly disparity, you're capturing disparity that might be on other dimensions that are part of that bias, right? How do you kind of, I mean, I think it's awesome. I think, you know, you can probably fit the bias, but how do you guys think about like that challenge?
So actually, one of the things that I skipped in the interest of time, there was one cluster that I sometimes show where you see a group of monarch butterflies that seem to be different from all the other butterflies because they're somehow dark. And then we looked at the images and we saw they're just underexposed.
Well, it's not about the image quality. No,
But it's just
Just like in terms of where, what the coverage. So there's two things, right? Like I think worth mentioning. There is the infrastructure development side of things, which is slightly dissociated with, like, it's a complementary but not associated with March's project with in terms of the work. So for the infrastructure, it's easier to think of like this as an intermediate layer between these portals and the user.
Totally.
And you're just serving functionality as it is. But for March's project, there will be a direct effort of actually control, trying to minimize those biases in terms of, like, for example, it's not unfeasible for butterflies to guarantee that we're going to have one image per species, right?
Yeah, totally.
Like, it's totally within the realm of possibilities. So we're going to have to do some concerted effort to not just rely on ID biodata. And actually target things more explicitly once we know exactly what we have. And there will be, because there's the bias in terms of like what kind of coverage of species, what kind of geographic areas, whether it's dorsal, ventral, and all of that. And we're just kind of slowly building systems to kind of do, but there will have to be an explicit effort of addressing that.
I just want to say, I think it's super awesome, and I think it's so exciting and powerful. But I also think that's a frontier area that is going to be so exciting and powerful as well to explore. Like, there are ways to, I think, control for that statistically and in sampling rates and stuff that is really great. So I just think it's awesome.
We're a minute behind on time, but there is a question in the Q&A that I would like to address before we move on, because they won't have the opportunity to continue, so.
So question for the last talk, have you looked into incorporating data layers from multi or hyperspectral imaging that includes reflectance or even just RGB values in your butterfly morphospaces?
Can you repeat the question?
Sorry, there was a multiple question here. Sorry.
Oh.
Sorry.
Sorry.
Click on the Q&A so we could read.
Yeah. Have you looked into incorporating data layers from multi or hyperspectral imaging that includes reflectance or even just RGB values in your butterfly morphospaces?
So this would be, great question. This would be fantastic to have. So right now we're kind of limited with what we download from the online databases, but there's actually, the plan is to potentially go in and take some more images ourselves in specific target groups, morpho butterflies with reflectance are obviously a good target. And we're also talking to some of the curators at the different collections to potentially integrate that. But that, I think, is further down the line. But it's really, it's of course important to also consider other spectra. Yeah.
Thank you so much. We have thanks.
Hello, everyone. My name is Neha. I'm a first-year PhD student at MIT. I work in the computer science department, and I work with Sarah Beery. So today I'll be talking about data sharing promises and pitfalls. So kind of as we know, data sharing happens everywhere, as we've seen from the talks yesterday and today. You know, different museums are sharing data with each other, different countries share data with each other. And in really the last 20 years, ecologists have made the transition from kind of keeping data to themselves and it being more of a like siloed model to changing to, this more open source, open data sharing. However, this doesn't happen without its pitfalls. I'm gonna really, the type of data that we work with is slightly different than what we've been seeing in the talks today. We work with camera trap data, so I'm going to be working off a case study of that. So, next slide. Sorry, I keep clicking away. You should be able to do it now. Okay, great. So let's say that we have this example data sharing setup across the country of Africa. We have 7 different national parks. Each different national park has camera traps that have been placed within it. Each of these national parks also has a different distribution of species and a different number of images. So in this case, when we think about data sharing, we're kind of asking the questions like, should Inankishu be sharing their data with Serengeti or vice versa? And should these national parks be sharing it amongst themselves? And if they do it, like what is the gain that they get from sharing their data? And in particular, we're looking at this from a machine learning AI lens, like what is the gain that they're getting from sharing their data if a model is being trained on like all of the data versus just their own? So, and also like, that's within Africa itself, but there's also the question of what happens when outside institutions ask for data. So if we're not even looking at these national parks, what happens if like the Kew Institute in the UK asks for data? Yesterday in the discussion, we started talking about the CARES principles and indigenous data sovereignty. And this is where these kinds of questions start bringing up, potentially these national parks in Africa would not want to share their data with a country that has historically colonized them. And in particular, in the US, there's similar cases where tribal nations don't want to share their data with the government or certain academic institutions because of historical malpractice with the data that they've shared. Sovereignty is a big thing that we're looking at. And there's also additional concerns, which are, privacy concerns, in particular with camera trap data or different citizen science types of data like an iNaturalist. There could be human privacy concerns that we also have to take into account. There's also environmental concerns as the amount of data that we all have has increased sort of exponentially in the last couple of years with digitizing and camera traps and everything else. Storing that data takes an enormous amount of energy to cool the data centers that keep all of it. And finally, acquiring representative data is often expensive. As we're talking about GBIF, we're seeing that most of the data that is in GBIF is in these like North America, European countries. And if we're trying to get data from other countries like Africa, like South America, getting that representative data is often quite expensive and prohibitive. So then this kind of brings up the question, when and where should we share our data and when is it actually useful from a machine learning perspective? And we're not trying to say, don't share your data at all. Obviously, it's very useful for institutions to get access. What we're trying to say is that when sharing natural and in particular ecological data, we have to kind of take all these concerns into consideration and think about carefully when we should actually share it and when it's useful for machine learning. So we kind of ran this simple case study across the African national parks. I'm bringing up this image again. And basically what we did is that we said that, okay, let's hold out 2 test sites, Inankishu and Kalahari, and let's train a model on the remaining training data. So these other 5 national parks, and let's keep those 2 national parks from before as the held out test sites and see what is the most useful data that was there for those 2 held out test sites. And we found a couple of large trends that were helpful. So, local data is more helpful than global data. In this case, when we're testing on Inankishu, Serengeti was the most helpful training source. And that's because, it's closest to Inankishu. The species within were quite similar as well. Like many other things, backgrounds were similar, habitats were similar. So oftentimes when you're thinking of when these institutions are thinking about sharing data, maybe it makes more sense to share locally rather than globally across the whole country. And then second, which is maybe a surprising result in machine learning, which is that more training data is not always better. The ML community traditionally sort of says that you should always share your data no matter what, and more data is always better. But as we've seen, with the butterflies, and in this case as well, there are a lot of species which are quite visually similar. So these like top 3 rows, the top is antelope, the second is sort of large terrestrial birds, and the third is sort of like canines or large cats that tend to come out at night. So a lot of the images end up looking similar. And those are the ones that models often confuse the most for each other. So if, for example, in your national park, there's no examples, or there's very few examples of Gemsbok, it doesn't really make sense to add a lot of other antelope because the model will just start confusing them for each other. Yeah, and so next, this kind of led to this larger question of, do we need to use all of our data for a very specific task? As we're looking at these larger collections of data, we have things on the scale of like millions and hundreds and thousands of samples. But do we really need to use all of it if all our model is doing is like a very simple task like butterfly classification? It doesn't necessarily need all of the other insects, or it doesn't necessarily need the whole taxa to be able to do that. And then that brings up the question of, okay, so, oh, that's an interesting animation. How do we select the most relevant data? So here, if our question was finding antelope on the prairie, then how do we actually select for the most relevant data of antelope on the prairie from these larger collections? And in doing so, the work that we're working on right now in our lab is creating a benchmark specifically for this. So it would be specifically for sub-selection for task-specific model specialization. That's kind of a lot of words, but basically what it means is that, most days, now that we're working with larger models, ChatGPT, Dino, Segment Anything, like these models are quite big and they've been trained on millions, billions of images. And afterwards, to specialize them for a particular task like butterfly classification, we have to do fine-tuning. So training again on like a smaller sample of data. And the question that we're working on is, how do you select for the smaller sample of data to specialize a very large model to your very specific task? So if you have this training data on the left and you have your evaluation data, which is different possible tasks, which all have different data distributions, how do we select from the training data which is best for fine-tuning? So we're working on different methodologies. Some of them come from like clip similarity embedding, some of them come from different embedding space distances. There's multiple different methods that we're looking at. But kind of the reason why I'm here today and the reason I really wanted to interact specifically with this community is because we're working on this benchmark and we worked on it for several different domains. So one of them has been camera trap data on the right. And we've also worked on microscopy, histopathology, medical imaging. Like the problem of fine-tuning for model specialization isn't a unique one to a particular domain, is the point. And we're kind of looking to add more. And we'd be really interested in adding more from the the natural history space, from these museum digitized data spaces, like working with something like DigiBio, and trying to see, whether the same problems that we're seeing across many different modalities of data and across machine learning, whether the same methods can be applied to the space as well. Yeah, thank you so much.
Awesome. All right, hi everyone. I'm Eddie. I'm a PhD student at MIT, advised by Professor Sarah Beery, and I'll be talking about our work in enabling novel scientific discovery with multimodal AI. There we go. So for over 100 years, ecologists and biologists have been collecting images of the natural world to better understand species, environments, and complex relationships and how they're changing over time. And now, smartphones have enabled scientists to scale up data collection through community volunteers with an exponential increase in data in recent years. For example, iNaturalist, which needs no introduction to this group, has enabled this collection of over 170 million observations. And this has already let us do a lot of really amazing science, such as species range and understanding migration patterns. But there's so many more stories that the images on platforms like iNaturalist can tell. For example, take a look at this picture, which in iNaturalist is labeled simply as a zebra. But there's so much more going on here. On the right side of the image, you see two zebras who appear to be fighting, possibly over territory, food, or mating dominance, which gives insights into zebra social behaviors. On the left side, we also see some cows. That's interesting because it can help us understand how livestock farming is displacing wild grazing animals. Being able to efficiently search or query these image data sets has so many different scientific use cases. For example, we can understand or raise awareness about the effects of pollution by looking at hermit crabs using plastic waste as their shells. Looking at elephants, zebras, leopards, and other wild animals on roads can tell us about how human infrastructure is affecting animal movement and animal behavior. But there are so many more use cases, some of which we might not have even thought of yet. Our goal is to build interactive systems where we can make it easy for scientists to get any information that they're interested in out of these large data sets to enable new types of scientific understanding and discovery. We want to use multimodal AI systems to enable scientists to search through this data with something as easy as just a text description, which for all these examples is enough to find really interesting images for all these different types of ecological hypotheses. So let me walk you through an example of how such a system might work. In this case, I'm going to be using a multimodal embedding model, and this is a pretty simple direct approach. So let's say a scientist wants to search through iNaturalist's millions of observations for images which happen to contain a California condor tagged with a 26. This is an individual condor that's been tagged with an identification marker placed by ecologists to more easily track its movements and whereabouts in photos. So for each of the images in the data set, we'll be using an embedding model like CLIP. These embeddings store the semantic meaning of each image as a compressed representation. Then we can use the same multimodal model to encode the semantic meaning of the text also into an embedding. This type of model is trained so that the text embeddings and image embeddings with similar semantic meanings are going to be close together in that embedding space. So now all we need to do is compare the text embedding to each of these image embeddings to get the similarity score. And that score tells us how related the text query is to each of those images according to the model. And then we can just simply sort the images by their scores to get the images that are the most related. And hopefully if the model works well, the most similar images will be the ones that are relevant to what we want to find. In this case, we want the most similar images to be California condors with the green 26 tag on them. And so to finish up, we just return some of the top matches. But we can go further than this. This type of search with pre-computed clip embeddings, as I think some of our other presentations today use as well, are really, really fast. But clip sometimes has a lot of noise. It's not a very big model, and we could do better. So let me show you an example of how we can do that using large multimodal models like GPT-4o and Lava, some of which you might be familiar with. For example, for these two queries examples, we can see how some of these top retrievals mostly are not relevant to what we want to find, right? For the condor example, we just get one relevant image. For the owl feeding behavior example, we get no relevant images. So what we can do is take some of these top images and use a multimodal model to re-rank them. Basically, these models like GPT-4o they're really large and they're really expensive, and it's really impractical to run them on your entire multi-million image data set. But instead, you just take your top retrievals, maybe like 50 or 100 examples or so, and you only need to run your model and ask it about those images. And for those images, we literally just ask, does this image show the query? Then we use that result to re-rank those images. And in this real example, now instead of those top 9 images having just 1 positive, 6 of them are positive, and that's much better. And this type of ranking not only works really well, but combines the best of the speed and near instantaneous retrievals from CLIP with the power of these large multimodal models. So this isn't just some theoretical framework I'm telling you about. This is a real system which we built and deployed. And here's a quick video showing our tool in action. So we can put in our query, and in just 1 second, we get all of our matches. And of course, it's not perfect, but we see that a lot of these top images are what we're looking for. And this is searching through 5 million images, literally a second to get what you want. And I think this is incredible. You can automatically look through just massive amounts of data, in this case, to find images of a specific condor that community volunteers happen to have taken a picture of. This is opening up types of research that never would have been possible before. And to finish up, I'm going to give you a sneak peek of some of our upcoming work, which we're really excited about, because we think that while building demos like this are cool and they can be really useful, if we really want to be scientists about this, we need to figure out a way to evaluate how well these systems actually do at performing these types of automated queries. So I want to introduce everyone to Inquire, which is our new benchmark for expert level text image retrieval, and also the iNaturalist 2024 dataset, which to those familiar with INAT 21 and INAT 2019 and other ones, is a larger updated version of 5 million research grade images, all with Creative Commons licenses and taxonomic labels. So Inquire consists of 200 expert level queries, each comprehensively labeled over all 5 million images of iNaturalists, really making use of those species filters to narrow down how many images we really have to label to make it tractable. For a total of 24,000 relevant matches, or about 120 per query on average that you want to find. To build this benchmark, we interviewed a lot of different experts from domains, including ornithologists, marine biologists, entomologists, people involved in forestry. Queries were also sourced from academic literature, and they're meant to be hard. They're meant to be something that even our best AI models will find really difficult to demonstrate their visual reasoning, domain knowledge, and contextual understanding. Very quickly, here's show you that these queries cover lots of different types of categories, hopefully showing to you that this goes far beyond just species recognition and into lots of different interesting and challenging domains across tasks like species, context, behavior, appearance tasks. They also cover a broad range of taxonomies. Here we can show you the grouping of these queries by different iNaturalist iconic groups. And I'm not going to bore you with all these numbers, but just quickly, I want to show you one chart, which shows how the best models we currently have perform. And the one takeaway that I want you to get from this is that even the best model that's currently available, which is GPT-4o, it's making good progress, but it really has a long way to go before it's able to answer all these queries really well. This is still an open research problem with lots of interesting and different directions to improve. And our hope is that progress on the Inquire benchmark is going to correlate with progress in having these AI systems actually be helpful for scientists. So I'd like to thank my collaborators who I've listed here. And if you have any questions, please feel free to ask questions now at this Q&A session or reach out to me. Thank you.
A few minutes for questions. I was just going to open up the next. Julie?
Yeah, so this is a question for Eddie and also a little bit for Arthur, because you both showed these really cool like search query interfaces backed by AI data. And working with MorphoSource, I spend a lot of time thinking about search interfaces, and there's a concept from digital humanities like a generous interface that sometimes thinks that a search bar is a slightly ungenerous interface because it requires a user to sort of know what they're looking for, what they want to see before they type it. Whereas, it's like visitors to the Peabody, they don't go into a big empty room where one person is waiting who's like, what would you like to look at? And I guess but you have all this phenomenal structured data in the background. So my question is, how do you think we can use this to move beyond the stingy search bar.
If I had more time, I would show you some more examples that I have, but I think our long-term vision is that it wouldn't just be a search bar. It would be almost like your conversation with ChatGPT. You could provide examples, you could provide images, and the response wouldn't just be like, here's things that are maybe related to what you want, but the response would be some kind of complex script, like output with code that it would run to process and like look at these data and actually make these sort of complex observations and bring back like a report. So that's like, I think in the long term, we would really want to see.
I was just gonna follow up on that. Is there taxonomic, are there clear taxonomic winners with this? Is it that birds do really well and insects? You have no, you know, you type in cues and the insect, the AI just can't handle arachnids for whatever reason because it's an alien, you know, type descriptors.
So I think I haven't investigated it like thoroughly by the taxa, but I've noticed that a lot of the most challenging queries are ones that are related to this really fine-grained understanding, which does happen a lot with taxa like spiders or like butterflies, where you really need this fine-grained understanding of these differences to really understand what the species. Mammals comparatively tend to be easier, even for like an untrained person.
And the descriptors are more kind of commonplace and you know, a beak is, you know, no one's talking about Ramphatheca, you know, typing in there, but maybe they are, I don't know. Some people are.
I can tell the naive clip, when it comes to bryozoans, they can do absolutely anything.
Right.
So I have a question for both of you. It seems that we have these great tools now in place that can help science. Like you can find the condor and use the geo information to figure out where it has been over the last 6 months. But it's also an open opportunity for poachers and malicious behavior. And so it seems like as a community, we have a trade-off between, you know, how do you protect that data, but how do you have that data? And we want both cases, right? The scientist wants to know where the zebra have been, but you certainly don't want the poachers to know, you know, where certain species are. So I don't know that there's I'm asking you a question, but I think it's a difficult answer, but you know, if you guys have any thoughts or anyone else in the room.
Yeah, I think I do have an answer. My understanding is from what a naturalist does, when there's a species that's potentially sensitive to like human interaction, like from poaching or other things, they usually mask the geographic location to within like a much larger frame.
But for what you're doing, you want that geographic, so my naturalist, then the scientist can't use the condor location. And so we do need the data there and not there.
I think a naturalist has a really strong philosophical bent on making sure that they're not causing more harm than good. And so I think that's kind of locked into their system.
Right, but then for the scientists, how do they get that info?
You can try to request that.
It can be a special member.
Yeah, Hebert's the same way. We mask the data publicly, but for permission, you get the data. I have a question about the sharing data, and I would think, so maybe maybe this is counterintuitive for me, but to train a model that's really good at detecting zebras, I would think that you would want a whole bunch of different backgrounds with the zebra in the foreground. And so limiting the geographic scope, I think, would lessen the effectiveness of the model, or am I misunderstanding?
I think it depends a lot on like what you want. It's like, yeah, it just depends on how general you want your model to be. If you want your model to be, I want it to be really good at zebras, then yeah, you should have all of the diversity of background possible and all of those zebras. But if you're saying, but then at the same time, there could just like you add in a little bit of added confusion when you add in that diversity versus there's the question of, oh, do I want a really specialized model per camera trap? It's like, I'm really good at detecting zebras in Serengeti. And then you could work with the larger general zebra model at first, and then you could specialize that model to each camera. Right. So it really just depends on how specialized your task is and like what data you're expecting.
That's great, thanks.
I have one more question. Yeah, maybe this isn't so much issue for iNaturalist, maybe it is, but like like I work a lot with sharks, and for example, blacktip shark, you look online and there's often mistakes. This is true in iNaturalist and many other web systems, or a green sea turtle. And so that leads me to wonder about the underlying accuracy of the image database. It's the part of the problem is, people are gathering so much data, right? Thousands and tens of millions of images of animals all around the world. And, maybe there's an underlying inaccuracy rate of 10, 20%, let's just say. And the problem is the more we get, I'm just wondering, how do we count that? Or is there any thought in the community how to deal with these kind of error rates about misidentification? Is that just considered just noise in the system that's unavoidable? Or do we have some mechanical turk going in there and just fixing it?
I guess in herbaria, we have a problem with misidentified taxa a lot, or unidentified taxa. So like using these TSC plots is a really effective way of finding those sometimes, assuming it can accurately describe them. So I think we're getting to the point where we might be able to address those misidentifications. It's like our data should be grounded because it's in an herbarium, it should have a taxonomy, and sometimes it's not correct. So, and if you have enough correct data, it should be possible to.
What about for species that really doesn't differ by genes, right? Genetic data. So you might have two species that look just identical, but they're different species because based on DNA sampling, you're not going to, no computer is going to find them. No. Okay, that's interesting to know.
Can I comment on that real fast? I mean, at one level, those are real problems for like conservation oriented questions that might have to do with like defining how endemic a species is or its effective range size, right? Or at another level, like for some of these analyses where you're trying to find out phenotypic variability, the fact that they're genetically different and probably different species would be less important. I think it really depends on what you're trying to do with these data, what kind of inference you're trying to draw. So I think like it's really dependent. But I think the problem is cross-cutting. It's not a naturalist. Like we've looked at a naturalist error rates for identifications versus like herbaria, and you can't really exactly compare them. They're kind of apples to pears comparisons, but all of our data have error rates. That's just a reality.
I mean, yeah, I think that's, I was just thinking like, let's take a shark example, silky shark, bronze whaler, bullshit, they all kind of look the same. So unless an expert went in there and sat there for an hour, and even then probably would. So this is primarily species identification. That's what I'm thinking of.
I mean, I think that also touches a bit of the biological issues. What is a species, right? I mean, when we looked into our butterfly data set and we zoomed in on some genera, we saw there was some clusters were well isolated and others there was just tons of overlap. And then we even saw that some of the color species, some of the points were definitely where they shouldn't have been. So there have been some mistakes during the identification, which I guess we talked a bit about this yesterday, that there should be, maybe it would be great if there would be a way for us to feed back that information and inform the collections. So I think this is definitely one thing that would be great talking about more. But then on the other hand, definitely like this, what is a species, right? Like we look at clusters and then experts like you can go in and, look at a range of images, something like that. I don't know what the solution is there, but it's, yeah.
Do we have time?
Yeah.
Yeah. No, I just wanted to touch on the subject that, collections have the misidentifications throughout, right? Like I think, for example, I work a lot with bryozoans, so I reflect on the group a lot. It's like, I don't think I've ever been with a collection of bryozoa that is not, for certain groups, a mess, taxonomically. And I think one of the things that we can, maybe we need to abandon the idea that these tools are going to tell us what the species are and embrace the idea that these tools will help us make sense of the data. They'll help us identify cases where there's an anomaly, right? Like there's something that's classified as something, but they don't cluster together where they should. So then the system would actually benefit us in terms of actually making sense and processing the data that sometimes is backlogged. So just to give a concrete example, the Maguire Center for Butterflies has 8,000 drawers of butterflies from Florida
that are unsorted. So it's material that's never been looked at. And part of it is just like the amount of time it takes to contact taxonomic experts to come visit the collection, make sense of that data. On the other hand, if we can make those digitally available and we have tools that kind of do us like pre-sorting of those specimens, then perhaps we can accelerate that process and actually mobilize more data and ensure consistency of data across collections that doesn't happen nowadays.
Yeah. And auto triaging type thing, just working out what the worst case, you know, and the areas that need the most
attention. Yeah.
I think these are good points. I do think there's a way for machine learning to actually solve this issue. It would have to be more specific. For example, you know, say a photo was taken in a particular reef and someone knew that reef. And they knew, well, there's none of these other sharks in that reef. That data, that information could go in there. So the specificity could be built with the model. It's just going to have to come from experts who maybe wouldn't be ones designing software. So I think there's a way for citizen science to build into this. I'm just suggesting that, you know, if you just look at image on iNaturalist Scholar, there's going to be a ton of errors, but there's ways to fix that even for species that look identical, because local experts who know the area, you know, you might have some local diver who's just lives in that reef, works in that reef, and like, I know these species. That's definitely not X. So I was just trying to think of this issue of like, if we could have a system for fixing error rate, it would be fantastic.
We are unfortunately out of time, so hopefully this can continue later. Alex Atkinson is up next. Thank you all.
Okay, I'm going to talk about a slightly different use of neural nets, I think, than has been discussed for creating 3D scenes from capture in uncontrolled environments with consumer grade equipment. So why is this worth exploring? What are we interested in here? Being able to capture these unconstrained subjects in the wild that might have very complex morphology, that might even be very difficult for photogrammetric techniques previously to be able to reconstruct surfaces from or models from, and then to figure out if this is measurable and accurate enough for a biodiversity study use case. So I'm sure a lot of you are very familiar, as we saw yesterday. Photogrammetry requires quite a bit of setup sometimes. You need pretty regular image distributions and distances and focus and things like that. But this pipeline is built on top of sort of the first phase, the correspondence search, essentially feature extraction and key points and sparse cloud building, and then builds on top of that with a neural network that does a few different things I'll talk about in more detail, but it allows for a less kind of controlled capture environment as opposed to photogrammetry. So when you're taking photogrammetric models, we previously did some research with Austin Mass and Nicole James, another collaborator on this project, exploring how detailed we could get with photogrammetric plant models captured in the field. Again, we needed a lot of control to be able to do this, and we still found that around this kind of complexity here in this chart of A to B, you start to get somewhat significant errors in the geometry, or in the case of subject A, you cannot recreate a 3D model from even a really good photogrammetric capture of that specimen. But when it comes to these methods, you are able to reconstruct even these highly complex subjects and create kind of this nice, beautiful render. Another kind of benefit of this is that it stores these models here. This is not a mesh, it's actually just a render. It is the neural net itself that represents the 3D scene. And that then tends to be a much smaller data set. So that's stored kind of as a series of coordinates in like a JSON file and a few other files that tend to be around 200 megabytes. So it's pretty efficient. It works in a quite different manner when you kind of break it out. You take those first kind of two steps essentially of structure for motion, and then we apply a neural net that takes in the RGB data, camera intrinsics and poses and directions, and then outputs just color and density and renders that in a real-time rendering scenario, which then you can apply some measurement tools or mesh extraction to later. Just to quickly kind of show the differences here of those two approaches, there's two ways to render that are kind of leading the field right now. NERF or neural radiance based is based on ray tracing algorithms. These are traditional computer graphics rendering methods. And then on the other hand, we have 3D Gaussian splatting, which we're finding to be a lot more efficient. And as you can see in this kind of heads up, this is the same exact data set. You get a lot more environmental data captured from the 3D Gaussians and a little bit better visual information. And those files are also even more compact. And it's just much more efficient to render it. So you can use less GPU power to be able to render the same data with a little bit higher clarity. Yeah, so there's a few differences that I'm starting to mention here that the Gaussian splatting rendering method is a bit more lightweight. It doesn't have to sample the rays every time you want to produce a novel view. It's able to just kind of store that information and render a scene from a series of kind of ellipsoids that contain densities of pixels, essentially, as opposed to having to trace rays of light from any given camera and sample points along that ray, which is a very computationally intensive method of rendering a scene. It can perhaps render quite a bit more detail, but you're kind of trading off on that computational cost. So in our heads-up comparison, it's maybe a little unfair because we're comparing those across the same GPU. If we had a much nicer GPU, probably those NERFs would look a lot better than the Gaussians, but again, you're kind of at a computational cost that might not benefit you depending on your use case. Another nice thing about the 3D Gaussians is that because those Gaussians can kind of overlap with each other, you can actually get an explicit surface representation. So I'll get into this a little bit more in a second too, but we're trying to extract models, 3D meshes, traditional triangular meshes from this, which are not necessarily implicit, especially in the NERFs. But the way that the Gaussians are rendered, you can get that explicit surface representation. It's a little bit easier to extract. And just to kind of clarify this difference too, it's a render, it is not actually a mesh. So you're able to do things like this, like pass through a subject. The render will show you any even impossible view from the compilation of images that you have in that data set. Okay, so just a little bit on the back end. This is all kind of run on basically Linux command line and like Python scripts. So right now, there are a few apps, Luma and Polycam, that are kind of emerging and doing this pretty well, but those are sort of like in a black box kind of state. So you can't really edit parameters or change how many faces you need to extract from the mesh, things like that. So you kind of get what you get. So you have to kind of get a little bit into the weeds a little bit on like Linux command line and editing scripts and things of that nature. But and then just on the hardware side, you need at least 24 gigs of VRAM and you need CUDA-based chip architecture, which means you need NVIDIA. NVIDIA is driving a lot of the research into this. We're using cloud-based solutions to be able to handle this processing. Yeah, AWS, and we've got 2 A10 instances. We're going to go up because we want to test those more computationally intensive NERF-based methods. We're not really able to extract any of the NERF-based meshes right now because it's that 24 gigs of VRAM is not enough to process that information. So again, very computationally intensive. And then what we're really interested in, again, is this surface reconstruction. So getting that traditional mesh, it seems like it seems likely that people will start to build into the renders ways to do analysis. I think Polycam kind of has a rudimentary measurement tool built in, so that's starting to emerge. But obviously, traditional meshes are what people tend to work with. A ton of tools are built around that. So we're exploring how to get good meshes from this. There's a ton of methods right now. We're testing to figure out what method of NERF-based rendering and then mesh extraction is going to work out the best. We've got some leads, but kind of still open in that way. As far as the 3D GS methods are concerned, Sugar surface-aligned Gaussian splatting for efficient 3D mesh reconstruction is definitely leading the field in that. So yeah, we're trying to figure out, is this good enough for morphometrics and landmark analysis? So kind of traditional stuff. And we're finding that we get an accuracy of around 10 to 100th of a millimeter, depending on the size of the specimen. I think this one is maybe about 2 inches across. And yeah, so then just kind of to bring it back towards photogrammetry, This is on a downsampled data set, 720 pixel across for the images. And you can see just that heads up, sugar is able to maintain vastly more geometric clarity here than Metashape on the exact same data set. And something that we were really focused on was kind of this moment here on the occipital arch that's like a little bit broken. You can see obviously Metashape not able to capture that. Here's the 6K resolution images that we eventually fed back into Metashape as well. You can see there that arch, and even on that full resolution photogrammetric reconstruction, it still doesn't quite maintain that accuracy. Here's a zoom in of our sugar-based mesh extraction, and you can see it's able to maintain that super fine geometric detail. And that's really one of the benefits of these methods, is like extremely fine geometry isn't really a problem for it. It's able to maintain translucent surfaces, reflective surfaces, things that are really challenging for traditional photogrammetry. Actually kind of can help these algorithms because the way that they're sampling light in a scene and reconstructing things based on reflectance in the scene. So some of those things might actually help. So yeah, some of your data sets that maybe have failed in photogrammetric pipelines might work out great for these. Here's just kind of the textured heads-up texture comparison. Again, the texture and the render is a little bit of a misnomer here. It's not a traditional texture. This is like a mesh with an overlay of that Gaussian rendering on top of it. So it's kind of strange. Again, like you can export a mesh with a texture, but you're kind of having to sample it from these different rendered views. So it's definitely a little bit different and a little bit wonky at the moment, but again, emerging kind of very rapidly and advancing as well. So yeah, back to that question of can we create these kind of like accurate models from less than ideal field conditions with consumer grade technology. This is a video that I took. I'm sitting in the back of a kayak in a river, the Winchester River in Florida. And this is a pretty bad capture of this specimen. Photogrammetry, this would very likely fail or produce like a pretty poor model. And then here's what that render looks like on a 3D Gaussian based render view. And you can see it's pretty photorealistic. It's doing a great job of capturing a lot of detail in the specimen and even in the surrounding environment. And here's another render method that we're exploring. This one seems to be rather promising in the NERF, neural radiance field oriented stuff. Baked SDF is the algorithm name. And yeah, you can also see that you can get a lot of depth data as well. So again, in reconstructing that surface geometry, that's pretty important. Here's the extracted model from that same capture. Pretty decent given the conditions. And yeah, just some takeaways here that it shifts your costs from maybe storage and capture hardware to your computational infrastructure. So a bit of a trade-off there, smaller file sizes. You can even use smaller image data sets, but you need a lot of computational power, which may or may not end up being cheaper in the long run if you don't need to store all the extra data. So it's a little bit more efficient depending on what you're doing. It can work with these more incomplete data sets. So like that dandelion, maybe it can revive data sets that were perhaps considered useless before. It can do unconstrained capture. It doesn't need that regular distribution of images and distance. And you can use consumer grade cameras. And again, it's got that fine elements, the complex geometry, reflectance, translucency, things like that are not really a problem for it. And it's decently accurate at the scales that we've been working with so far. So, yeah, and our acknowledgements here. So yeah, thank you to IDIC Bio and the NSF for funding this, and Austin as well. Yeah, cool.
All right.
Lovely.
Take it away.
Thank you. Hello, everyone. I'm your dinosaur interlude for the day. This has very little connection to a lot of what's been talked about, but I do have plenty of digital data problems, so maybe you all can help me and I can give you a dinosaur break. A lot of the work I do is in the paleontology space. Yesterday, someone said paleontologists measure bones, which is not inaccurate. We also do a light touch of
Suspense.
maybe. We also do a light touch of Jurassic Park vibes. So a lot of our work is kind of somewhere in between that space. My research focuses on reconstructing the motion of extinct animals from fossils and trying to figure out how we can do that. The issue is the data we have to work with looks a lot like this. I didn't realize drawer was a scandalous word until yesterday, but we go to museum drawers, we see the fossils, and we're trying to figure out how these animals would have moved. The issue is that I often say a lot of paleontology for centuries was vibes based. It wasn't really based in data. And so paleontologists would pick up fossil bones, not sure why that's moved down there, that's interesting, but they pick up fossil bones, they kind of move them around in their hands and they try to get a sense of what poses seem possible or seem impossible at a joint. And then they'd come up with these representations of those data, trying to say, we think the joints can move about this much, so this animal probably moved this way or this other way. They'd put this data on bar graphs and they'd kind of throw their hands up and say that's the end of the day. So a lot of my work has been trying to adopt methods from 3D animation and computer animation and apply those things to paleontology to make these reconstructions more data-based, more reproducible. One of the things we did is we took character animation joints, which are what underlie animated characters like Sully, and we applied a series of rotations to those things to try to see what all the possible joint poses in a joint were. And then we can use those same techniques from computer animation to get the computer to say, at this pose, the bones are inside of each other, so that wouldn't be possible. So in this sense, we're able to kind of standardize these reconstructions, make them reproducible. But we still have that vibes-based element because when we pick up bones and move them in our hands, we're trying to get a sense of what's possible at joints. We're not just asking ourselves if the bones bang into each other. We also have a sense of whether joint poses look right or look wrong. And we also need that layer brought into our reconstructions. So paleontologists have tried to do this in different ways. If this is the neck of a long neck dinosaur, like the one in the galleries, they'll look inside the joints and say the bones overlap by a certain amount. And so we think the bones need to overlap for the joint pose to be real. That's clearly not the full story because if you look at the knee poses at the bottom of the screen, they all overlap by the same amount, but some of them still look right and look wrong. So the vibes aren't bad, they just need to be formalized so we can apply that in a real scientific way. The way we do this, if you're looking at the ankle joint of a bird, these are living dinosaurs, so we can start here. They do the silly thing where they fuse up the sole bones of their feet. So you get a really simple joint. That's just two bones coming together at a bird ankle. And if you zoom in on that ankle joint, we can focus in on the joint surfaces that actually interact. And we can use different tools in that computer animation software to think about how these surfaces interact in 3D. So we can shoot a series of little vectors from one surface to the other following the curvature of the surface. And we can measure things about those vectors like their lengths and their curvatures to describe this joint interaction. If we look at this joint pose, it looks pretty good, but if we slide the bones apart, that's where we get that low overlap condition that we know is bad. Now we have a way of measuring that. We can also maintain overlap, but move the two halves of the joint different distances apart. And that low symmetry condition is also something that looks wrong to us, so we can kind of detect that. And finally, even if we maintain overlap and symmetry, if our bones hit each other at a funky angle, we get in this low curvature congruent situation that we also know looks wrong. So if we take those three kind of parameters, that formalizes our vibes or our intuition for what a good or bad joint pose is. And using tools within computer animation software, we can formalize that into a quantitative score for a given joint pose. So you can see if we do this for a ton of different 6 degree of freedom configurations of those two bones put together, instead of just saying this looks good or this looks bad, now we can assign a quantitative score to that. If we do this for a bird, we won't get into the details of the space, just think of every little dot to the joint pose that your joint could assume. If we only went in terms of bones banging into each other or not, we'd have to consider a ton of different joint poses, which is more than any animal would ever use. But by using these kinds of scoring parameters and looking at the 3D of the bones and how they interact, We can get beyond this binary possible, impossible, and we can assign an articulation quality to every single joint pose and think about the differences in how the bones fit together. And we can start to kind of apply thresholds to think about what the highest scoring poses in that joint space are, which is cool because we've gone from this vast expanse of 1 billion possible poses this joint could use to a very small sliver of that potential pose space. Now, all of this is with living birds. We need a way to validate this data. This is my first digital data problem. We end up creating these really cool animations of living animals walking on treadmills. I can't get into the methods of this. I'm happy to talk about it over lunch. But the way we do this is through taking high-speed X-ray videos and combining the data from those videos to create 3D animations. Right now, all of our X-ray videos live on a specialized database portal for X-ray video footage because there are no physical specimens that go with these data, so repositories don't like to accept them. But our funding is not sustainable for that. So if anyone has a home for our terabytes of X-ray videos that I swear are super useful, please let me know. What's cool here is we have a bird walking on a treadmill, and the ankle poses that it's using are actually falling within that highest scoring region of pose space. So just from the shapes of the bones, we were able to figure out how this animal walked in life. And this applies across tons of different speeds, across tens of thousands of frames of these X-ray videos that we've taken, and we can see this is a consistent relationship. This is that bird walking on a treadmill in a standard video, and we've gone from the shapes of just its bones to this real life behavior. So we did this for a ton of birds. That was a guinea fowl. That's a polka-dotted bird you saw. We did it for emus. We looked at some recently extinct birds, and we kind of got the confidence that we could apply this to dinosaurs. So moving on out to a dinosaur. This is a dinosaur Deinonychus. Over lunch, if you head into the collection space, it's like the famous Yale dinosaur. You should check it out. She's right against the back wall. It's this famous raptor from Jurassic Park. It's also in more recent series, and we now know they have feathers, but they're these really cool animals. What's exciting about them is they have these giant killing claws on their second toes. So Deinonychus means terrible claw. And what's exciting about this specimen from the Peabody collections is we can laser scan it, get 3D models of the bones. It doesn't CT well. We can talk about that later. But you have all of these different joints that make up the foot. Of course, we deposit these data in MorphoSource. Ta-da, MorphoSource. Nelson controls my data. I had never met Nelson before this workshop, but Nelson controls my data. What's great about vertebrate paleontology here is we love to make these things fully open download, and MorphoSource lets us do that and publish them all with our papers. So now if you want to see the killer claw of Deinonychus, you can go on MorphoSource, check it out and download it. If we run that kind of an articulation joint analysis for all the joints of this animal, just based on bones banging into each other, we wouldn't be able to eliminate much. But if we apply those scores, we can get down to a really small region of pose space for each of those joints. And then we can apply the kinds of thresholds we found from living birds to this dinosaur to reconstruct how this animal would have moved in life, assuming its joints operate under similar conditions. With this, we're able to hand off these data to professional animators who make nice, fancy animations. And that animation is featured in the kiosk right in the collection, so you can check it out when you head back there. The final thing I'll talk about is we can also take a look at that killer claw in more detail. There are a ton of hypotheses for how the animal would have used it in life. And we can apply these same computer animation techniques to ask, well, if in a slashing, killing stride, the animal would have had to move its claw like that, what happens to the joint articulation score throughout that? And we think it's dropping too low in slashing, so it's probably stabbing its prey instead. All of this to say, this is how we get from our drawers to computer animation. So it's a fun application of these digital data. The final question I'll leave you on before I sit down is my other digital data question. I know we have folks from Cornell and other places here. A lot of the times we can apply these methods to living animals that are endangered as well. So we have a similar project looking at the helmeted hornbill, which is endangered because it's being hunted for its task material. We like to publish these things, and we have scans from museum collections, but oftentimes it's great to publish images or videos or sound recordings of these animals in life as well. And we have a really hard time figuring out how to search for images and recordings that we're able to publish that have the proper licensing. So if anyone can talk to me about that over lunch, I'd appreciate that too. Anyway, that's your dinosaur interlude. Thank you.
You know, that dreaded talk before lunch. Be aware of that. Two beautiful talks by Alex and Armita. It's gonna be hard to follow that. I think, do I just press the middle button?
Sure.
Oops, I'm going backwards. It seems to be, sorry, I'm seem to be flipping through the wrong
I
way.
think if there's two arrows. Arrows,
Oh, I see.
I'll put you back to the beginning.
So just to provide a little background, it's been wonderful to listen to these talks. I've really enjoyed it. I'm looking forward to meeting and talking to you all after lunch or during lunch, but I come from a little bit of a different background. I'm a biologist. I work with animals in the field. I work with sharks, marine mammals, sea turtles, many reptiles, amphibians. So I'm interested in using these 3D tools because I don't come from that background to understand morphology and the locomotion and the conservation of these animals. And so I'm going to be talking to you about some 3D work we're doing with little conservation to look at body shape and body condition, and some new 3D photogrammetry tools to visualize morphology in the field. So the goals are, first off, I want to talk about a paper that's just been accepted into Methods of Ecology Evolution, will be accepted probably. A new 3D photogrammetry system that can be used in the field that can create extremely high resolution 3D models of, in this case, bird bills from live animals. And I think it might be interesting to talk to you all about that. And the second is new machine learning approaches that we're about to submit a paper on for estimating body condition in marine megafauna. So the first part, my collaborator from University of Washington, and Josh Medina, who's my PhD student, Alejandro, because he comes from Colombia, got to pick the name, which is Picocan. For those of you who speak Spanish, I think you'll understand it. Picar. And this system was designed because Alejandro came to us and said, we would like to visualize hummingbird bills while live. We can't kill them. And when you have a hummingbird that's been in a museum for a long time, or any bird, the beak deforms. It can deform, actually, according to him, even in a matter of hours once dead, because there's moisture in the beak. And the species that he works with, if ever you ever have a chance to work with or go to Alejandro's farm, his finca, it's incredible. 8 species of hummingbirds just flying around. It's truly amazing. He's able to capture them and hold them. But then the question was, how do we actually visualize the beak? Because this beak, these beak differ among these different species, and he wanted a way to do that. So Josh, together with Josh Alejandro, we came up with a photogrammetry system that tried to overcome a lot of the early problems that we encountered in some of our own 3D photogrammetry system. One of the big problems that's been discussed here is with 3D photogrammetry, it's very hard to do with live animals. And it's what tends to happen is you tend to have tons of cameras that becomes incredibly hard to travel with. If you ever tried to travel through Colombia or Southeast Asia with 50, 60 cameras, as I've done in the past, it's a huge problem. So we wanted to get fewer cameras, a higher quality, higher resolution system. So what we ended up doing was building a system that used a combination of lighting and camera systems that was able to focus on at a very high level macro photography to these objects. So this is the system. I'm just going to walk you through some of the basics. If you have any questions, I'm happy to talk about it. But you have 6 cameras. You can have more if you want. They are the Sony Alpha 7 II that has a really good macro, 100 millimeter macro. We have a system here that allows a focus on a small object, something like the size of a hummingbird beak. And we use depth focusing to get a really clear, crisp image of these beaks. Once you're able to do that, we're actually able to get really high quality scans, less than 0.2 millimeters accurate. So to give a sense for it, if you have a small screw about this big, is able to visualize the screws behind that. And I should say, I want to give the bulk of credit here to Josh. I had built a similar system, the BeastCam technology. It was not nearly this good. He's the first author in this paper, and it's really his very impressive technological capacities that allowed this to happen. But we're very excited about this because if you actually look closely, I always thought hummingbird beaks were just these beaks, but they're not. Here's the fur, but here's all the little grooves and nooks and crannies on the animal. And you start to also understand, for example, the sharpness of the tip. There are differences among hummingbird species, the sharpness of the tip, and we're able to quantify and visualize that. And the beauty of this is it will take these hummingbirds, get 3D scans in the beak, and then let them go. It's fantastic. So in total, we gathered 3D scans on 18 different species of hummingbirds. That shows here, and these are actually these hummingbird beaks. These are some of the 2D images of the 3D scans. These are actually shown to scale. So if you look at the top one, that's Endostheria. And these actually all occur, by the way, in this region in South America. But you can see there that this species has quite a bit of variation in the size of the beak, but you can also see some of the tremendous variation in the shape of the beak, anywhere from very straight to very curved. There's tremendous variation in sharpness. For example, this species, Acanthorix, has an extremely sharp beak, and that's because the hummingbirds, just like dolphins, are jerks. They jab their beaks into each other and get what they want. They rip out feathers, and they're very mean. And so there's tremendous variation there that has ecological value for these animals. We're able to extract very good morphometric data. So that was one of my main interests as a PI, was can we get good data from these animals while live? And we can. So these are different species, length, width, height, volume, and surface area. So this was very exciting for us, and we could see tremendous variation among species that was repeatable. We confirmed this with also the measurements that we took with individual calipers. The second part I want to mention is a project that we're about to estimate body condition in whales. IV body condition is really just for humans who are trying to lose weight typically. For most wild animals, they're trying to put on weight. And for a marine mammal or a shark or sea turtle, essentially the more mass they put on, the more they can reproduce. It's actually one of the most biggest parameters that impacts reproduction, longevity, fecundity, everything in a marine mammal. And the other, obviously, issue is when you look at a marine mammal, there is the Marine Mammal Protection Act, which makes it a felony to even disturb a marine mammal. So we have to use non-invasive, clever approaches. So this is a collaboration with Frederick Christensen from Aarhus University, Subransu Maji, and Shandak Bagchi. I want to give them both the credit to Shandak. What the approach that was really built off what Frederick Christensen had done, except he had done it really by hand. He had an amazing database of drone images. These are southern Atlantic whales. This is in, I think, the images from Argentina. So we can get excellent images. The water quality is amazing. And then what's kind of cool is the animal turns on its side. And because this animal essentially is a giant hot dog, we're able to basically visualize this 2D image ultimately to a 3D extrapolation and create a 3D mesh and 3D volume. And fascinatingly, how do we know this is correct? We know this is correct from historical whaling data because people killed these animals in droves, slaughtered them, and then actually measured each body part. They knew the weights of every little piece. And so we're able to go back through history and confirm it. So we actually knew even from these two images, if a whale simply rolls over, even if it doesn't in some cases, you can actually very accurately estimate body mass. The problem was you had to do it by hand. And that was very time consuming. So that was the case for machine learning. So with this machine learning algorithm, it first off was used a masked CNN approach, which basically identified different animals. So we have a mother and a calf here. It measured length because we had rangefinder data. And then ultimately created this 3D system of sort of chopping up the animal like they used to in historical times, and then extrapolating that 2D to 3D. We used approximately almost 9,000 images from unarmed, unmanned vehicles, drones essentially, off South Australia. This is Frederick's data set. And from that, we were able to actually create a model that is initially we're very happy with. It's approximately 93% accurate for body volume mass and 99% accurate for length. I do believe this can become more accurate with more data, better. New technology is always evolving, as you know, with machine learning. So this shows, for example, the model generated body volume. This is the manual measure. So we actually have data manually compare it to that. And that's the distribution of errors on the right. Now, the other thing I've been doing that I won't talk about today is we've been doing a tremendous amount of work creating 3D models of many living animals, sharks, marine mammals, and putting them out there for people to use. I'm actually excited about this machine learning to actually just recreate these animals. I hadn't thought about it that way. And the reason I bring that up is that when we look at this image, these are some of the sharks we've reconstructed from a great white to a small leopard cat shark. We have data showing that in terms of body volume and surface area, you take one, you go all the way to the other, identical. Their ratio is rock solid the same. And that's actually true, by the way, all the way from a snake to a whale. So surface area volume relationships seem to be extremely well preserved. And so with this machine learning system, I wanted to actually use this to ground truth the 3D meshes, because up to now we've actually had to simply go in and film individual animals, and it's very time consuming and risky and so forth. And with this system, we believe we can speed that whole process up. So sorry again to be that talk before lunch, but thank you very much. What's that?
There's still more here.
I know, but I'm still before lunch. So anyway.
So we have about 10 minutes. If anybody has any questions. Please speakers. Sorry.
So I actually have a question for Armita. So I was curious because how you initialize, like, because it seems like you do some kind of exhaustive search of the perimeter space to then kind of find the scores. So I was curious how you initialize that and whether it was exhaustive or it was actually some kind of iterative.
There's nothing iterative currently, it's fully exhaustive. So yeah, for joint configurations, you have rotational mobility and translational mobility. So right now we start at 5-degree rotational resolution and 7x7x7 grid of potential translations. And we kind of focus in on the highest scoring area, and then we rerun the process at higher resolution. I am interested in picking up iterative approaches. For this initial study, we did it fully exhaustively just because that's how paleontological analyses have proceeded so far. They're really just checking everything to see what works. But I think there are definitely smarter ways to search this space, and I'd love to go into that.
Yeah, because I was curious, it seems that if you reflected one from the top to the, like, its opposite side, but you could use some kind of image registration approach as people just to kind of like see which one would give the best fit between.
Yeah, so that's a interesting part of it is this assumption that you're searching for the best possible fit, but because these joints are moving, there are actually several poses that are kind of equal quality or equal fit throughout, if you think of your finger joints throughout like a flexion extension cycle. But I think there should still be search approaches to get at all of those things. Yeah.
Related question, how about consideration of soft tissues, particularly with your modern samples?
Yeah, that's an approach we had looked at previously, is reconstructing and modeling soft tissues and their properties. But the issue is that's just layers upon layers of assumptions, because in the fossil record, all you often have to work with is the bones. So the benefit of this latest approach is really taking the data that's directly preserved in museum collections and not having to make assumptions about soft tissue properties and letting the shapes of the bones give us the majority of the information. And so when we found that match to what living birds do, that was with no assumptions about soft tissues whatsoever. So we're really hoping to stay true to the data that we actually observed.
Great. We have a Q&A question, I believe, for Alex.
Yeah. Alex, are you finding difference with how NERF or Gaussian perform with data sets generated via using a turntable camera combo, camera stationary versus camera in the hand / moving around an object, camera moving?
Can you read that again? I'm sorry.
No, you're good. Are you finding a difference with how NERF or Gaussian perform with data sets generated via using a turntable camera combo, so the camera is stationary, versus a camera in hand moving around, so you're physically moving the camera?
Yeah, there is some weirdness, and it seems to vary from algorithm to algorithm. The Gaussians tend to respond well to the turntables. Some of them fail based, but it sometimes works. So it kind of varies. I think if your background is actually too consistent, that can be a challenge for either algorithm. Yeah.
I was gonna ask now, so do you notice any, have you looked at color quality between the Gaussian? I mean, I know there's a lot of model creation things, but the fidelity of color?
No, not yet.
Okay, do you just anecdotally, do you think there might be a difference there, or?
They're both really just working with RGB data at the end of the day. So I think it's going to give you almost exactly what's in that image. It's going to come back out. So as long as if you color check your image or whatever, I think it would probably be a 1-to-1. But I haven't. Okay.
Seeing no other questions. Matteo, can you go ahead and share your screen with us?
Yes. Can you see it?
Yep, we just need it. Yep.
Okay.
All right. Thank you so much, Matteo. Take it away.
Okay. Hi, everyone. Sorry, I'm not there in person, but I am in a moment where I am trying to transfer to Baltimore in just 3 weeks. So you can imagine the kind of hustle that I am at. Anyway, today I want to make the argument that museum collections are really important for biomedical research, especially when we want to include an evolutionary framework within our hypothesis. So my research mainly focuses on phenotype, which is the response between the relationship among genome and environment. But the reason I am so obsessed with phenotype is because morphology per se is the only bridge that we have to correlate modern diversity with the extinct one. So morphology per se is the only language that we can read to reconstruct the history of life. And I do this with different sources of data. I use obviously the fossil record that represents the extinct species with modern species, where modern species are both intended to be the adult morphology and the embryological one. I usually apply data collection that goes basically in the field to collect eggs or to collect fossils. And then I use a bunch of 3D imaging techniques. We will go through over them later on in the talk. And then I try to quantify the change of morphology across ontogeny and evolutionary patterns in order to create a holistic framework and create a holistic hypothesis. Or at least I hope this is what I'm doing. Now, the problem with different sources of data is the fact that not all of them are carrying the same kind of information. So the fossil record is basically just skeletal morphology. We know that there are biomarkers and some genomic data, especially in fossils that are more recent. However, their availability is very restricted. So modern species are the key because they can tell us two things. One is what is not preserved in the fossil record, such as ecology and behavior. But at the same time, the plasticity of embryos can give us information regarding the mechanisms behind what takes to change morphology per se across evolution. And the reason for that is that embryos all start in a very similar manner, at least from a morphological perspective, at the beginning of development, and they change through development, becoming the adult morphology that we know. So is this a recapitulation of evolution per se? Obviously not. But what we can do is to create an evolutionary framework of developmental series across different species and start to check what are the small changes that happen early in development and that get added to the morphology per se until they diverge to adult morphology. So in other words, we can change what are the we can check what are the changes that define, for example, the presence of feathers or the presence of a beak, just comparing the developmental trajectories that differentiate between a bird, a mammal, or a reptile. Now, this is easier said than done. And the reason for this is because although we would like to check mechanisms, this doesn't mean that we have accumulated through the last decades enough information to understand them. Indeed, when we look at developmental biology, most of the work that has been done has been focused on just 5 species among vertebrates, when we know that there are more than 70,000. And when we look at the diversity of species alive today, there are different estimates between 3 and 100 million. Obviously, it's a huge range of difference, but still it tells us we are far from characterizing the morphology of the species we know, and especially their development. So from a molecular perspective, so when we look at the genome, there are several initiatives that are happening around the world, and several methodological innovations that are helping in understanding the mechanisms per se. So CRISPR-Cas is one of them, and now we can edit the genome of species we know the genome for, and understand what is the correlation between the change in the genome with the morphology. Another way to look at gene expression and how the genome is shaping the embryo is spatial transcriptomics. So at this moment, we can check gene expression in 3 dimensions within the 2-micron resolution, which is pretty unprecedented. And obviously, there are several initiatives in order to sequence all the genomes of modern vertebrates and life in general, such as a 70K genome project. But when we look at morphology, we have been pretty much behind. And for a long time, this has been the state of the art for imaging embryological samples. So we use alizarin red and Alchan blue, and I'm sure that in your museum collections, you have quite a bunch of samples with this kind of staining. But as you can see, basically, this is staining are focusing on the skeleton per se and the formation of the skeleton through different stages. Now, the problem with this staining is that we are missing all the early stages of development where the skeleton is not appearing at all yet. And the second thing is that Alchan blue is basically staining cartilage, which happens late in development, and alizarin red is bone per se. So we don't know basically anything regarding early development, and especially in those species that are not part of those handful that has been studied for a long time. And so, as you might already guess, this is far from creating an evolutionary framework that can connect development with biomedical studies and evolutionary biology per se. So during my PhD, actually at Yale, I decided that I wanted to focus on imaging morphology of non-model organisms. And you can do this in several ways. One of these is synchrotron, especially for the smallest samples, where you don't need specific stains, but you can image at the subcellular level the entire embryonic sample, which is great if you want to track cell movement and cell differentiation. The downside of this technique is that you have enormous files. So in this case, this opossum head is more than 3 terabytes of size. So in the long run, is not really applicable on the broader range of collecting data across a tree of life. The other easier way is CT scanning, and I'm sure you are all familiar with this technique, where you can use iodine stain or PMA staining if your museum manager or collection manager allows you to. And what is interesting about this technique is that it's reversible, at least for iodine. So you can go in the museum collections and actually collect data that would be impossible to be imaged just a few decades ago. But what is important is that you are not only collecting skeletal information, but also soft tissues. And here I am reporting a couple of examples of some primates come from the Field Museum, where the embryos were still preserved in the collection within the placenta and the uterus. And so in this case, we are collecting information regarding the embryo itself, but how it's interacting with the adult female. So you can create these kind of data sets that are not only related to morphology, but also how morphology is made. But a technique that was developed quite recently is clarity and immunofluorescence that is applicable to non-model organisms as well. And basically the technique was invented in Stanford in 2013, published in 2013, where you can get a sample, a biological sample, and then you can clear out the lipids, making the sample transparent. And at that point, you can apply antibody fluorescence and target different systems through development in order to image this in 3 dimensions through confocal imaging. And so this is the result of one of these data sets. So in this case, we have a zebrafinch embryo that was stained for early skeleton and muscles. And what this tells you is that we can image multiple systems at the same time within the embryo, and we can apply this kind of tissue differentiation and imaging throughout the development. So we can start from the beginning of development, like in this case, a little skate and a cat shark, when the embryos are incredibly similar to each other, and then track small changes that eventually will lead to morphological differentiation. And so in this case, you can see a stage 31. So it's basically a fully developed skate where you can see the head right here. These are the gills, and these are the wings or pectoral fin. And the images we can take have both tissue level resolution, but also cellular level resolution. So in this case, we can track cellular differentiation and system differentiation. So once you apply this to the vertebrate tree of life, you can actually range from the early gnathostones, in my case, with the little skate and cat shark, up to birds and mammals. And in this case, you can actually build a broad evolutionary framework that will help us understanding how development is affecting evolution and how the biomedical studies that we are so keen in expanding are actually affecting our perspective on evolution and medical implications. Obviously, because these data sets are in 3 dimensions, what we have the opportunity to do is to understand how morphology changes in 5 dimensions through the application of quantitative comparative methods, where obviously you have 2 time dimensions, which is ontogeny and evolution, and the other is the 3-dimensionality of the morphology itself. And so with this, I want to thank all my collaborators and previous advisors, and obviously all the fundings made this work possible. And I want to thank you for your attention, and I'm happy to answer your questions.
Okay, we'll have 2 other presentations, and then we'll open it up for Q&A if you can stick around. Thank you so much. Share the sound this time.
Okay. Good.
Should be.
Okay, well, thanks, everybody. This is fun. I have to say, this workshop is blowing my mind. Rob and I were just commenting a couple of minutes ago about the things people are doing. You know, several years ago, these iDig Bio workshops were about maybe we can digitize things. And now they're like, look at what we can do now that we've digitized things. It's truly amazing. I keep waiting for a boring talk. I haven't seen one yet. You know, and I can't believe I saw a dinosaur walk. 3D renderings. I mean, this is so fun. But now I'm going to give you a break because I am going to say nothing new, nothing you haven't heard yet before. And and in two ways. One is I gave actually a version of this talk at a previous workshop, but I thought it fit into this workshop better than the previous one. So I decided to give a version of it again. And everything I'm going to talk about today has come up in some of the previous presentations. And so it's kind of fun to see how everybody's converging on similar things. But I'm going to do it at very much a 30,000 foot level and not get into the gory details because I don't understand any of the gory details. I'm just going to talk. This is sort of my view of what some of this digitization and bringing machine learning and AI tools to bear on those digitizations, sort of categorizing some of the things that we are already doing and can be done. So I'm the director of the Macaulay Library. So the specimens we deal with are very, very different from the specimens that most of you deal with in your collections. We started with media, right? But it was analog media. And because of the material we work with, we got into digitization about 20 or so years ago. So we started digitizing very early on to turn these analog media into digital media. And now the collection is very large. We have millions of photos, audio recordings, and videos. But there's specimens, you know, because Ted Parker recorded this musician ran at this particular place in this particular time, it is a specimen. It represents the phenotype of that individual at that place and time. And just as the physical specimens you all deal with do. So we started digitizing analog media. A lot of you have been digitizing physical specimens. And the important thing is that once you've turned those specimens, whatever they started as, into a digital specimen, there's so many things you can do with it. And that comes through, you know, here's my complete understanding of AI. But once you bring those tools to bear, I think there's sort of different categories of things that we can do. So one is curatorial issues having to do with data quality. There are tools and enabling people to use the specimens for science and for conservation. And then there's things we can do with those digitized specimens for engagement and education. And I just want to give a few little quick examples of each one of those things. Again, most of these examples have already come up in more detail in previous presentations. But first, a little bit about the kind of digitization we're doing. In a nutshell, for our collection, what we're digitizing, or what we're not digitizing, what we're using machine learning for is to identify and classify. So you find birds in pictures and then classify what species they are, or find bird vocalizations, recordings, and classify them to species, right? So in a nutshell, that's what most, but not all of the machine learning work that we're doing that Macaulay does. It starts with people like Taylor here, who know a lot about birds and vocalizations and can listen to these recordings and annotate them. And she knows what all the species are, not only in the foreground, but in the background, draw bounding boxes around them, identify them. And so we develop training sets that way. Same thing for photos. We can put bounding boxes around birds in pictures and use that for training data. And we rely on a community of bird experts out there to do this annotation for us. So at any given point in time, we have about, three dozen or so people helping annotate these recordings, and we're using local people with local knowledge about the birds in their area to do these annotations, as Glenn mentioned yesterday. And then all that is fed into the machine learning algorithms. Grant Van Horn has been working with us extensively, as with Sarah Berry, and he's also working with iNaturalist. Sam Heinrich is on our staff. And Grant and Sam and others have been developing these models to do things, right? So now all of these different things that they're doing are all kind of based on some of these same models. And this is a video, sort of Grant playing around with a video detection and classification, and then on the bottom, vocalization, so identifying everything in the video, and then the audio to species. Right. And so we've taken that and developed this thing we call Merlin Sound ID. And it's pretty good in some regions of the world for identifying vocalizations of birds. So North America and Europe, it performs extremely well. South America, India, not quite so well. Australia, very few species in the model. It's a personal embarrassment to me. That's where I do most of my research, and it doesn't even work there. So I have to explain a lot to my friends and colleagues down there. But you know, we're getting there and continue to train these models and expand. And so now that's the kind of machine learning work we've been doing. Now I want to talk about applications of that to these different categories. So first, for curation, for us, and this came up actually in one of the previous presentations, data quality and in particular identification, misidentification of specimens is a huge issue because most of our data are being contributed by citizen scientists who may or may not know actually what they're listening to or seeing, right? And so actually making sure that the identifications are correct is a very serious issue. So that's, and so what we're doing to address that, and this is in process, we sort of had a beta version, we haven't really incorporated the way we want to, but we are trying to incorporate these machine learning algorithms into the data submission pipeline. So when somebody uploads an image or they upload an audio recording and they say it's whatever species, the algorithm helps identify whether their identification is correct or not. And this is very important to us because just last month in May, we had nearly 1.5 million photos uploaded. We had 78,000 audio recordings uploaded. Nobody can listen to that. No human can listen or look at all those photos. We have to have algorithms help us out with that. So we do, this is the prototype. Somebody uploads a bunch of pelican photos. They say, okay, these are all brown pelicans. And then the algorithm says, well, wait, this one doesn't look like a brown pelican. It looks like a, white pelican. So it very gently nudges them. You think maybe you might have misidentified that one. Maybe that's a different species. Same thing, we can do the same thing with audio. So we're in the process of incorporating these algorithms into that pipeline to help with the data quality. The other second category is science and conservation, actually helping people use the digitized material for whatever research or conservation purpose they have. And there are really two ways of doing that. One is helping researchers find the material they need. When you have millions and millions of photos, the joke I always say is the bigger that haystack gets, the harder it is to find the needle in the haystack, right? Especially because most of these data that are being contributed by citizen scientists are broad and massive. There's lots of data, but they're very, very poor in metadata. It's usually place, time, species, right? And so anything else that might be captured in the photo, it's almost never in the metadata. And so Grant and Elliot Miller and a couple others at the Macaulay Library got interested in this question. I wonder if we can train models just to go in and find pictures of nests or pictures of eggs so we can get an idea of when do birds breed. For most species of bird, we don't actually know when they breed. This is very parallel to the earlier talks about plant phenology, this bird phenology, when are they breeding? And so they took all the photos where somebody had actually said there's an egg in the photo, trained an algorithm, ran it on the collection and found thousands more pictures where there was an egg in the photo that hadn't been labeled. So then we can go back and add that label to every one of those photos, right? We can tag them appropriately. And so, yeah, so this is playing with that. And then, but then the next step beyond that is, well, can we get away from using tags? And so this is where Grant, along with Sarah Berry's group and others, are like, can we use natural language, large language models to allow people just to query the collection in some way that's easy, right? And so this is the prototype. The Grant started, this is really a prototype of a prototype. It's a very early sketch of the natural language, but Grant sent this to us a few months ago. So ask it anything you can. And so I'm like, okay, I want to find all pictures of birds with yellow eyes. And so I came up with things. A lot of them just have yellow near the eye. They don't actually have yellow eyes, but it's doing okay. And just with further refinement, birds with long bills. And, I was actually pretty impressed. So with refinement, I think, and Eddie's talk earlier, I thought was great. And Arthur's talk, it's the potential for this to help people find the material they have that they need is massive. Like those are two very exciting talks, I have to say. Those are very cool. Now, the other way that I think machine learning tools are going to help the research and conservation community is allowing us to build tools that those people can use. Use our data to build the tools. So they don't need the raw data, they need the tools. And Glenn's going to talk about that in the next talk. So I'm just going to show this slide to tantalize you. And then last, I want to talk about engagement and education. Like all of these talks that we've been seeing at this workshop, such beautiful imagery, such cool, inquisitive things. These are hooks for engaging the public. And so our hook is really important for us because we rely on the public to contribute data. So we really need to get people enthusiastic about taking pictures of birds or audio recording birds and sending us that data. And so engagement is super important to us because we are a participatory science endeavor. And so we put out Merlin, which really was, as we were saying yesterday, Merlin is just a tool to get people excited about the birds in their backyard, right? That's where we started. And then we said, well, maybe we can add sound ID to it, Shazam for birds. And so we did that. And this is Merlin users per month. For several years, that dotted line there is when we added sound ID. And after we added sound ID, it just skyrocketed. So last month, we had 4 million people in May used Merlin. Really amazing.
I'm one of them.
Subtracting the people in this room is what's 20 short of 4 million. But I want to show this video. Allie Smith, who works at Macaulay, wondered what are people saying about Merlin online, and so she went into TikTok, and she found several hours worth of TikToks where people are talking about Merlin, and she just made this little short montage of people talking about it. And so to end, just because it's kind of fun, I just want to play this.
So I'm going to show you how to bird like a pro. And all you need is one app. That app is called Merlin. It's called Merlin Bird ID. The Merlin Bird ID app. Merlin Bird ID. Merlin. Where you just open this app and you hit sound ID. You download like whatever region you live in. And then it'll identify all the birds around you and light up when they sing.
Look it. That was just all in like 5 minutes of me sitting out in my back deck.
This app is now part of my personality. I just left a work meeting, heard birds chirping, hopped in the whip, pulled the app out. House finch.
I'm obsessed. I'm never gonna stop using this app. Like, I can't even handle it.
So I'm addicted to this, not in a literal medical sense, but I don't think I could live without it.
I'm addicted to this, so addicted. I'm addicted to this.
My dad sent me this the sound app for birds. And you press play and it listens for the birds around you and it tells you what bird it is and what the bird looks like. So now my ass over here, since I've been using it, thinks I'm like a bird expert.
It's the Merlin Bird ID app, and I cannot recommend this enough. I would never consider myself to be a birder. Never thought I'd see the day, honestly, but this app has really just full-on converted me. Every time I hear a little chirp, a little squeak, I'm whipping out the app, I'm trying to take a picture, I'm trying to find the call, I'm trying to call back to it. It's honestly really fun.
Okay, so yes, I heard of the Merlin app before. I use it all the time in my backyard.
lots of birds we have caught on it. We got a yellow-throated warbler, I think it was called. We get blue jays, cardinals, white-throated sparrows all the time. And yeah, it's an awesome app.
So I've just been listening to bird chirps on my walk, and you will be amazed at the number of birds you hear. I can identify blue jays. I'll come home and tell my husband I just heard a cardinal, robins. It helps you be more mindful as you're paying attention to what's around you instead of thinking about your worries or what your day was like, those types of things.
Hear the birds chirping. What kind of bird is that? My boo identifies it because she got the Merlin app. Is that a black and white warbler? Nah. Think again, the cinnamon colored feathers. It's a Carolina wren.
I just brought it down here and given it a go, and it's identified widgeon and red shank and black-headed gull and a great tit. And it's absolutely brilliant. It seems to work really, really well.
I want you to do an owl hoot. And I want to know if it really sounds like owl.
What's it say? Barred owl. Take that.
Okay, so that was just for fun, but there are a couple serious things out of that montage that I wanted to emphasize. One is that almost nobody in that looks like your typical stereotype of a birder, right? That's a broad audience. The second thing is what they're excited about is not really the birds, but they're being empowered to do something they never thought they could do, which is identify the birds in their backyard. And I would posit that the material that you all are digitizing, used in the right way, could lead to that same kind of empowerment. So that's what I want us to think about and frankly do. So that's it for me. Thanks.
Following up against like the internet's most entertaining bird personalities, like not really the act you want to follow. So just keep that in mind when I finish my talk. Just try and keep that TikTok over your head. It kind of Mike's closing remarks. Find it all right.
Maybe. Hold on. I thought I had it.
Just said Seeholzer.
No,
I put it in the Google Drive folder. Yeah, Seeholzer ML.
Thank you. One second. Here.
Share sound? Yeah. Okay. I don't have that many sounds, but. Okay, so I'm Glenn. I was part of ML. I've been at ML for about 2 years from background in physical museum collections. A lot of the stuff I'm going to be talking about here, I can't take credit for. I've been a lifelong recordist. But again, all the Merlin bird ID stuff, that's an enormous team from ML and this whole global community of contributors and users. So just keep that in mind. Everything in there is based off of that kind of global community. And so one of the principal goals of museum science is to document biodiversity, right? Oh, I also mentioned I'm really impressed by you guys that can do talks without notes. I can't. And I finished this a couple hours ago, so we'll look at these for a minute. So one of our principal goals of museum science is to document biodiversity. And we usually achieve this goal traditionally through physical specimen collections. And we've really come a long way in this discipline from like the Victorian era stamp collector mentality to this really rigorous scientific discipline that we've seen on display in the past 2 days. And I think we'll all recognize that over the evolution of this discipline, we're always finding new and exciting uses for specimens. And this is often aided by new technologies and new perspectives and new questions. How do I go forward? This way? Did I do that? Okay. And so this in general, I think this concept, I like to think about museum philosophy, this idea that we're going to be repurposing specimens that we collect for one purpose now for something completely different in the future. And that we should be taking a lot of care and thinking through how we're doing things now so it'll be maximally useful into the future. And I think we can have a lot of faith that this is going to happen because we've seen it happen over and over again. And so, and this museum philosophy I think can be seen in the expanded impact of sound archives, of reference sound archives, as an extension of the museum collection community. From their origins in the analog era to these kind of amazing uses, the amazing uses we're putting to, we're using them for now, like Merlin Bird ID and automated applications. And so I'm gonna go into a bit of the history of ML that leads us up to these kind of exciting applications. So Macaulay Library, as Mike mentioned, is one of the world's largest archives of digital media, and it's especially true for birds. That's definitely our specialty, though we have big collections of anurans and marine mammals. And so again, we're really rooted in the traditions of natural history collections. There's been a lot of just back and forth between physical collections and this archive throughout its entire history, which is about a century at this point. And so each one of our millions of assets, we consider a specimen documenting, the presence of that species in space and time, phenotype, behavior, all that stuff. And so our first recording, how would I play that recording? Would you do that? I think you should play right there.
LNS catalog number 16968.
Just listen to this for a second. So this is a recording made almost 100 years ago. It's the first recording in the ML archive, and it was made using this new technology at the time, synchronized sound and motion picture technology that you had to take down in a large truck and use this big parabolic reflector to get anything that was like useful. And I think we take for granted that whenever we listen to a recording, we're listening to the past. It's really cool to think about a century ago. This bird was singing in this willow tree in Stewart Park on a spring day. It was recorded by Arthur Allen. And so the equipment they used to make this recording was heavy and kind of like prohibitively expensive. But this early generation of recordists really saw its potential and invested heavily in the engineering to make this stuff more portable. And this was a game changer because up until that point, the only way we could document biodiversity was through specimens and where we could see the plumage, morphology, colors, maybe paintings, which might get us closer to some kind of behavior, and of course, descriptions. And so this is the first chance to document behavior in the wild. And so we started, and so we started that's how I do it. And so we started this museum collection of sounds with the goal of documenting the acoustic diversity of the natural world and using the same rigorous data standards as museum collections were beginning to employ around that time. And so over the years, as we developed the systems here, we really invested in the engineering behind recording technology and kind of pioneered advances in portable recording technology. So a big advance was magnetic tape reel-to-reel recorders that were 20 to 30 pounds, but for the first time, you could put them on your shoulder and go out in the field, and probably with an assistant with holding the parabola, get really good recordings in situations where you couldn't bring a car, or couldn't bring a horse-drawn carriage. And so the initial goal was to document, get a representative sound of all the birds in the world. And that was a huge, overwhelming task 80 years ago. And so to help complete that, it couldn't be done by one team at one university. You had to really expand the number of people that were contributing to this archive. And so there's a really active outreach to new recordists, people interested in technology, recruiting, training, to get recording units in the hands of people that were in really far-flung places, such as South America. And so I'll get back to that expanding community. But we'll fast forward to 2000, as Mike mentioned, once the digitization technology got to the point where we could have really high fidelity digital copies of this like a continuous waveform, we invested heavily in archiving, digitizing and archiving all of the hundreds of thousands of reel-to-reel tapes we had there. And you have to play each one of those reel-to-reel tapes in real time to digitize it. So it's just a huge, long process. It took us about 12 years to do. At the same time, transcribing all the metadata and then building technological infrastructure to house the stuff on the internet and send it out to the community and make it visible and playable. And so coming back to this expanding group of recordists, this was, I think, one of the early kind of citizen science projects, participatory science, getting people that weren't necessarily professionals involved in the discipline and contributing recordings. And so one of these like early pioneers of recording was this guy, Ted Parker, who Mike had on his other slide. He was actually a board member of the lab for a long time. And at the time of his death, he died quite young in his late 40s. He contributed 1/4 of all the recordings in the Macaulay Library. So this guy was a fanatic. He just like lived and breathed bird song recording and doing it in really, really kind of trying field conditions. And so he got his start actually in museum science. He participated in when he was young, early expeditions from Louisiana State University Museum of Natural Science, going and collecting expeditions from remote areas of Peru. And gradually his focus turned to bird recording, and in collaboration with the Library of Natural Sounds, as Macaulay Library was then known, he got the right equipment, and especially the kind of back and forth that allowed him to piece together this, what at the time was this almost impossibly complex wall of sound of the South American avifauna, and start really trying to put names to all these sounds. And in the process of doing this, he already had this, he built this encyclopedic knowledge of sounds, and he began to see the potential of using that to do surveys. So up until that point, it took months, maybe even years, for scientists to feel like they climbed that species accumulation curve and asymptoted, like, okay, we know what's in this locality. And he was finding he could do this in a week, 2 weeks, at least for birds. And that was huge. No one else could do this at the time. And so he was going out and putting this idea to the test and going to places that had conservation potential, were under threat, or just poorly explored, serving them relatively quickly, and then turning around that data and making conservation recommendations. And this is now, I think we take for granted that we can do these things quickly, but it had to start somewhere. And in the neotropics, at least, it started with TED, Conservation International. And these methodologies were later were employed by the Field Museum. And they really resulted in all these major protected areas. TED's maybe crowning achievement was getting the Medellin National Park created in Bolivia. The Field Museum has over about 20 years, turned Loreto, Peru into this network of reserved areas. And we all love seeing these nice shaded regions, knowing that there's some conservation significance or some conservation plan for these areas. But the data that's used to understand what's going on in these areas are really just pinpricks. We just have like little dots of information from these areas, and that's all we really know about these places. And we can see that at an even bigger scale with the eBird data set. This is essentially where people go and look at birds. And you can see that, lots of people are going lots of places in Europe and North America. But as you get into the tropics, and especially like right along the equator, you have these big gadadaps, Congo, well, especially Siberia, Indo-Pacific, and then in the Western Hemisphere, the Amazon. And so how can we go in and fill these data gaps? And so we all know that there's, there's an added importance to this data gaps at the moment. And that's these global bird population declines. So we look at South America, we all know it's a really biodiverse place. At any spatial scale you want to parse it, peaks of biodiversity in birds are going to happen in South America. And one of the really special places on Earth really is Yasuni National Park, which for not just birds, but across all taxa is, there's a general consensus that's the most biodiverse place in the world, and certainly the most biodiverse protected area. And so in that Yasin National Park is this place called Tipitini Biodiversity Station. It's been around for about 30 years. And there's all this really cool research happening on neotropical ecology. The whole point of the research station is to expand our knowledge of Amazonian biodiversity and use that to promote education and conservation. And so about 20 years ago, a research group from the University of Florida, led by Betty Wiesel and Don Blake, set up these long-term bird monitoring plots. You can say Puma plot and Harpia plot, 100-hectare plots. And they started monitoring, and their initial goals were community ecology. We just want to understand better how the community of birds in this place is assembled. But as they started sampling through time, they started realizing that probably the biggest contribution of their data set would be as an alarm. Not that we need more alarms to tell us, things are not going well. But what they found over 20 years is that the bird populations have basically been halved. Mistnet captures are have a decline by about half, maybe even more. And the same is shown, it can be seen in audiovisual observations. You just can't find as many birds as you used to be able to find in this patch of forest. This is particularly surprising because Tipitini is in Yasin National Park. There's been no history of any human impact, no logging, very far from direct human impacts, but this is still happening. And so our best tool for conservation that we thought of, protecting huge swaths of habitat to kind of be a reservoir to replenish all the areas we're messing up. Even that plan seems to not be working right now. And we don't know why. And we don't know at what scale this is happening, how many other places it's happening. We have some hints. Oh, and I'll just kind of let's see if this is gonna work. Okay, yeah, so this is a recording I made in 2009 when the bird populations hadn't really started declining. And that little piping sound in the background, that's not the focal sound, that's a black-faced ant thrush. And it's this terrestrial insectivore, and it just calls all the time. If it's around, you're gonna hear it, and it's gonna be a background of all your recordings. It's kind of annoying until you go back 10, 15 years later, and those birds are completely gone. So that species is functionally extricated from Tipitini and maybe from the general region. And it's not just that bird, it's this whole guild of terrestrial insectivores. They're basically extricated from Tipitini, and indigenous guides, local guides from the region basically report the same thing. All these territories, and these are often the birds that people go to Ecuador to see, to the Amazon to see. They cannot, their territories are unoccupied. They can't find these birds as easily as they used to. And so something big has happened, and we don't really have a handle on what, how big it is. We do have these clues, and this is a big plug for big bird plots. These are our best tool for long-term monitoring. We have so few of them, and we need to support them. There's 5 places where we have long-term data sets. 4 of them have been resurveyed recently. And in 3 of the 4, there have been major declines. Pipeline Road and in Panama and Manaus, Brazil, major declines primarily in these understory avifaunas. Cochacashu appears to be pretty stable, and that's actually encouraging and an interesting point of variation that may allow us to untangle what's happening. But these are just single points on a very, very large map. And so we need to we need some kind of complementary data set to get a handle at a larger spatial scale. And so what we're trying to do is this other type of methodology, this big population modeling that eBird and the Center for Avian Population Studies that Cornell has been piloting. And their goal is to go from these static range maps to highly detailed, high resolution range maps, and then eventually to these status and trends products where you can see at fine spatial scales where bird populations are increasing and where they're decreasing. But to get to that, you need lots and lots of observational data, or at this point, observational data. But as we mentioned, there's big parts of the Amazon of the world where birders aren't going to go. They're never going to go. At the point that they can access those places, they're probably already cut down and deforested, and we've maybe lost the battle. And so that big gap in the middle of the Amazon for the black-faced ant thrush is basically because there's no data from those regions. That's not an actual, that bird is there, you can't resolve it. And so what we're looking to is the passive acoustic monitoring methodologies, automated recording units, deploying them in these remote regions to get these baseline data sets that then you can then resample. But you can't collect all these big, massive data sets without the tools to analyze them. And so that's where Merlin Sound ID comes in. And the idea is that we're going to try to integrate these two streams of data, observational data and these acoustic detections, to help understand these bird population at a large spatial scale in the Amazon. And probably over time, so I'll just quickly run through the kind of 4 ingredients that we need. First and foremost, it's an improved reference archive of Amazonian birds. Basically, half of them just don't have enough recordings at the moment to train a reliable model. And then even when you have those recordings, you need to get lots of annotations. You need people who really know the bird sounds from these regions. And that skill set can take years or decades to build up. Then you also need to go out there and actually get the data from these regions. And this kind of, this looks like a big data gaps. It's actually even worse than that, because these are aggregations of points. When you zoom in, really where we're actually sampling is just really, really, really tiny little slivers of habitat in this vast, vast sea of forest. And then, and so we want to be using these acoustic monitoring units. And then also we need this like massive overhaul of technological infrastructure to house all these recordings, analyze them, synthesize that data, store the detections. And so we're off to a good start. We're collaborating with Tiputini, and we've run these recording workshops to help build out the group of observers and recordists in the Amazon that are contributing recordings. And then, yeah, that's probably where I'm going to end it. And I think just final plug that these reference bird archives are still the foundation of all these things we want to do for society. And I think that's a general lesson we can take for natural history collections.
Okay, and we just have a couple more minutes. I think we could take take a question or two if anyone has any questions for our last few speakers. Yeah, Ross.
One, it's worse, isn't like worse than, you make it sound like it's pretty bad. It may even be worse than that. Even still, like I kind of wonder about the interest-specific call variation across regions for different species, like how much calls vary within species across some of these.
It's not really like geographic variation that's the issue. It's more like just the repertoire.
Yeah,
the repertoires. These things are really can be pretty sizable.
Yeah.
Luckily, most of the South American outfawn are sub-Aussians, and they have learned they have like innate songs, and so they're relatively standardized. And so these models do pretty well in those. But then you have mimics, and I don't know. There's a lot of complications.
But I think it's a beautiful application of like the direct value of these resources for like conservation outcomes. That's a really good thing.
Yeah, we're hoping, the policy.
Okay, well, I would like to thank all of the presenters who attended this series, as Mike mentioned, and so many other people mentioned. It's been incredible to highlight all of these advances within the use of media and associated with collections and biodiversity. I've been with IDIG Bio for 9 years now and continue to be in awe of what you all are doing to increase our knowledge of the natural world. I'd like to thank Nelson Rios and the Yale Peabody Museum for hosting this workshop, and thank you to all of the organizers who helped to curate all of these great speakers and content. This has been about a year now of planning, and it's finally here and almost done. For those that are attending virtually, all of these recordings will eventually be posted unless speakers indicate otherwise. And I'd like to just thank everyone again for tuning in and contributing, and I look forward to seeing what you all continue to do for the future. So yeah, thank you, and thank you to all our speakers. And yeah, I have one quick announcement once we're done with. Yeah, thanks Matteo for tuning in.
Thank you, Zoom.